A very good afternoon to our revered guest speakers, faculty delegates from different colleges, and my fellow colleagues. I welcome you all on day 10th of the AICT sponsored 2-week faculty development program on exploration of nascent pedagogical tools for updating teachers of pharmacy education. Today, we will be having two technical sessions. The first will be taken by Professor Vandana B. Patravale and the second with Dr. Kanika Thakur. So, starting with the first session, today we have with us a pioneer in the field of education, a great researcher and an academician, Professor Vandana B. Patravale. She is a professor in the Department of Pharmaceutical Sciences and Technology, Institute of Chemical Technology, Mumbai, Maharashtra. A very warm welcome to you, ma'am. Ma'am will be speaking today on Six Sigma and Qbd, paradigm shift in development of pharmaceutical products. Though ma'am doesn't need an introduction, but still I would like to briefly introduce ma'am to everybody. As mentioned, ma'am is working as a professor in the Department of Pharmaceutical Sciences and Technology, Institute of Chemical Technology, Mumbai. With around 30 years of teaching and research experience, Dr. Vanna has over 200 refereed publications with an H index of 45 and I-10 109, 15 granted patents, 22 patents in pipeline, and 2 trademark registries. Her area of research includes development of nanocarriers with major emphasis on infectious diseases, cancer and neurodegenerative disorders, medical devices, development, nanodiagnostics and nanovaccines. She is a recipient of several awards. A few to be named are the Fellowship of Indian Chemical Society 2020, Kukreja Oration Award 2020, APTI's Dr. Manjushri Pal Best Pharmaceutical Scientist Award 2019, Shri Amrut Modi Distinguished Research Award 2018, OPPI Women Scientist Award 2015, and many, many more. She is actively collaborating with researchers as well as industries within the country and abroad and has completed Indo-Swiss, Indo-Japan, Indo-UK projects. Ma'am has transferred about 20 technologies to industries, including drug-eluting stunts being marketed in more than 60 countries. So this is a very brief introduction of ma'am's achievements. A very warm welcome again, ma'am, from Lloyd Institute, and over to you.
So, title today of my talk is Six Sigma and Qbd, Paradigm Shift in Development of Pharmaceutical Products, as Dr. Shilpa mentioned. And looking at the program, I did see that some part of it has been covered by Mr. Big, if I'm not mistaken.
Absolutely, ma'am.
Right? So
Seeing
it's
our sessions. That's so nice of you, ma'am.
Yeah, so I'm sure that he would have given you a real good inputs as to which are the softwares which are used, how it is to be used. But typically, what people are not able to answer at the end of any such lecture is when to use which design. So I'm sure today's lecture will give you some insights of it. So we will start first with product life cycle. One needs to understand this well in order to understand why is it that we are talking about Six Sigma and QBD. So if you look at the product development cycle, definitely initially you need to develop a prototype product. To develop a prototype product, your drug substance has to be in place with respect to its physicochemical properties. You have to set the analytical methods for the same and then look at which particular process you will utilize to design a drug delivery system. Once the prototype is ready, by prototype I mean that you do have 6 months stability data in place, you are moving ahead and doing tech transfer. So in technology transfer, you are actually doing pilot plant scale up studies. So your optimization actually starts right in the initial stages. But final optimization and validation is done at tech transfer stage. By process validation, I mean that you would be taking at least 3 consecutive batches using an optimized product process which you have already done, and assessing that whatever quality you had pinpointed right in the beginning, whether you are getting that quality or not. So, of course, once this validation is done, which is typically at 10x level, if I consider X as a prototype development stage, this becomes 10x. From 10x, you need to move to 100x, and that is actually the commercial manufacturing, wherein the product gets realized. You have to do periodic quality reviews. Obviously, once the commercial manufacturing is done and the product comes into the market, then also starts pharmacovigilance. And in case there are some issues, you need to deal even with post approval change management. And this process will continue because people would go on modifying the dosage form in a way that they get excellent quality so that there is no failure at all. Now, QPD majorly has been done to tackle this particular step of post approval change management. What was happening earlier is, because you had only finished product as a final QC, one had to redo all the exercise and submit all the documents to regulatory agencies. Now, because you are developing the product and building the quality right while developing the product, you are coming out with a concept known as design space. So, as far as you are moving within that design space, you don't have to go back to regulatory agency. And that is one major advantage which industry is deriving by following, of course, QBD. So this is a classical development approach wherein you have a fixed process. You already decided what process I am going to use, but in case you have any input variations, you are going to get a highly variable output. So just consider a tablet dosage form. You have decided that you would do the process which is direct compression. Now, in case your blending is not uniform, the particle size is not uniform, there are mild input variations, they are going to lead to tablets of variable hardnesses, tablets of variety of thicknesses, which will finally lead to dissolution related issues. So, what we are going to do is, by using Six Sigma and QBD approaches, we are going to control the variations. And you will see in the further slides, how are we going to control these variations. As you can see in this slide, it is very well depicted that the risk goes on increasing, the quality falls, cost increase, customer satisfaction also decreases when you have variations. So key to controlling failures would be to control these variations. So this depicts that age-old systems have been used. 5000 BC, we were just checking whether whatever we are using is fit to use or not. All trials taken, you would have read, people were using feathers of birds even to inject, in fact, certain parenteral preparations. So that was 5000 BC. But by 1800 AD, specification and tolerances came into picture. And it is only in 1924 that what you know as quality control parameters, they came into existence. So it is a iterative and reiterative process so that quality can be maintained. As you know, quality is of prime importance because we are dealing with human beings and every life is special to us. So, what is a new regulatory perspective? New regulatory perspective says that you need to control the materials which go in the process. You need to know absolute ins and out of the product you are going to process, and also know the processes in depth which will be utilized to manufacturing these products using quality materials, and therefore keep variations within acceptable limit. So now this has shifted. There's a paradigm shift. Now you have flexible process, so that even if the variations are there input, you do get constant output. And I'm sure you would be familiar with techniques like PAT, which are nothing but process analytical tools which would be used. We talked about blending, which can result in highly variable output. Now, in the blender itself, you can install probes, and those probes can be near infrared probes, so that till the right kind of blending is done, the process doesn't move forward. And therefore, you always get a constant output no matter what. If the raw material, there are certain slight changes, they will be taken care by the flexible processes. So now the new regulatory perspective has changed only here. You can see that you are making commercial product as per the control strategy. This has become a regulatory norm. So even if people were practicing quality by design earlier, till the time the regulatory guidelines came and it became a must, people did not start giving attention to it. So now we know that it is a regulatory norm, and product has to get realized as per the control strategy. To come to this control strategy, you need to use QBD. And therefore, this is facilitating continuous improvement. So the process has become, I would say, simpler. Now, what you can see here in shaded areas is I have highlighted the ICH guidelines. ICH guidelines Q8, 9, 10, 12, all of them deal with product development. And you can see that some guidelines are broad throughout, like Q9 is broad throughout. That means that guideline is playing role at each of these stage. Whereas when we look at Q8, that is very important at the product development, and then it tapers down as we move forward. Same way, Q12 doesn't feature till we look at commercial manufacturing. So it's very, very important. And my earnest request to all the teachers here, that in your spare time, please read all the guidelines of ICH. Unless we are clear about the ICH guidelines, we will not be able to teach the students what industry needs. And industry will follow the norms very, very critically. So, as the knowledge of these guidelines increases, we are decreasing the risks. That is the scope of continuous improvement. That is the scope of QBT, and that is the scope of Six Sigma. Now, what we will do is look at what is Six Sigma, and then whether whatever we are talking about QBD, does it differ? I say as a first sentence that both Six Sigma and QBD are enablers for getting the right kind of constant output. We'll see how. I'm sure you all are familiar with Sigma values, which probably we had done at B Form level. Some key facts about it. Typically, Sigma is a system of practices which was developed to systematically improve the processes by eliminating the defects. This is element of many TQMs, that is total quality management initiatives. But unfortunately, though lives of human beings is important, we have borrowed it from Motorola. That means this came first in automobile sector. And we have borrowed this technology from there. Whereas ideally it should have been in place for pharma first. Anyways, keep in mind that when we talk about sigma, the higher the level of sigma, lesser are the defects. Currently, our pharmaceutical industry is at 3 sigma level. That means there are 66,807 part per million defects. So we are getting yield of only 93.3% in most of the operations. And Six Sigma says that there will be only 3.5 or 3.4 ppm defects. This is also not 100%. You can see that it is 99.9997% yield. So there is still scope for improvement. Nevertheless, 3.4 seems definitely better in front of this figure, which is 66,807. So simply we can say that Six Sigma is nothing but a set of techniques and tools for process improvement. And this indicates where we are versus what we want to be. So we want to be as near the target as possible and still be within the specifications. And that is what industry desires, not this. We are within limits for most of the cases, but we are going out of specifications too. So this is our target so that we can reduce variability. So what is a methodology for Six Sigma? It is known as DMAIC, DMAIC, which is a short form of define, measure, analyze, improve, and control. So one, in industry as well as now we researchers should look at what are the customer demands, what are the market demands, then look at what data you have in literature, collect that data and keep it for future comparison purposes. Do cause and effect analysis and then see whether you can implement certain techniques so as to enhance in such a way that you hit the target. By hitting the target, we mean that you use the control strategies so that you can reduce the defects and therefore save more human lives than ever. So, in short, we are talking about improved processes. Again, keep in mind that this process is iterative and reiterative. Or you can see that this again comes to step one, goes through all the steps, and then again the process starts. That means people want to go to 100% quality level. We'll take one case study so that you understand. And incidentally, it is a case study of ranitidine hydrochloride tablet. And you know that ranitidine has been in papers for past at least a year and a half because of the impurity it contains, which is definitely not what is required. But this is a published data. So you will see that this will move as we move the slide. So first step is define. Identification of parameters was a must. Those parameters which are critical to customer include, of course, appearance of tablets. You know that ranitidine are coated tablets. And the coating, many times when you open, those of you who have taken ranitidine tablets as an antacid would know that many times you get that coating which is ruptured coating. So first step is appearance, compatibility, acceptability, whether it is to customer. As far as quality parameters are concerned, these are all defects which will cause batch rejection, which can cause batch rework, or even many times, FDA will take action and close your premises. And that has happened in past with many industrial units here in India. Not only in India, it has happened world over. So you do not want anything like that to happen. And what is it which is critical to process? An item If it is not held within a certain range as determined through a process development, can cause out of specification results. So we need all the step parameters of your pilot plant scale up, of your production to be understood well. You need to know what is a critical in process test, and you also need to know what are those critical important parameters within the process which you need to control. For example, again giving you example of blending, you know that the mixing time will be very important. You know that RPM will be very important. So will be what is the load which you have put in the blender. So likewise, for each of the steps, you have to know what are critical parameters. And if you control those parameters, I'm sure you will be controlling the variations. So this is the study of 40 batches labeled NL461 to NL500. And process which was under investigation was blending, compression, coating, and packaging. I told you ranitidine is a coated tablet which requires these major operations. So why is it that they did this study? Because they saw there were issues. So what was used for analysis? One needs to use or analyze the data statistically. So they've used softwares like Statistica and Minitab 14, as well as Microsoft Excel, and that has to be declared to regulatory authorities. So what is done? Once you discover that there are undesirable effects, look at what is the identification, what is the severity, why is that severity caused, due to which these undesirable effects are there. And then once you have pinpointed what is that severity, look at how can you make improvements in the process. So look at this process of blending, wherein I have indicated what are the assay values of ranitidine hydrochloride for all 40 batches. And you can see that there is a lot of variation in these. The frequency is not the same. So what did they do? Suddenly there was demand of ranitidine hydrochloride tablets which increased, and they were using the same blender right from the beginning. That shows that how important it is that you consider how much load is in the blender. So they changed the blender, existing blender, with a new blender which had higher capacity. So obviously for uniform mixing to happen, you need that blade should be able to rotate and mix the contents. And therefore, just by changing the blender, validating that particular system, they could control the assay to quite an extent. Moving on to compression, what did they realize? You can see that this is the chart. Even if the slides are not very clear to you, you can see that it gives average weight. This gives average thickness, average hardness, and friability. You can look at batch to batch non-uniformity. In fact, these red lines are the limits which are in-house limits. And in some of the cases, the results are going outside the specified limits. So you know that if thickness changes, definitely the hardness will change. Hardness changes, all other related parameters will change. Here in this case, one case, even the friability of the batch was failing. So to overcome this, what was their control strategy? Because again, suddenly there was change in scale. And those of you who are teaching validation and regulatory requirements would know that revalidation has to be done even in cases where you are changing the scale. Now, changing the scale meaning you may increase the batch size. It also means you may decrease the batch size. Both the cases ask for revalidation. So, if that is not done, you will see that such things would result. So, you have load on machine. Because of load on machine, there is wear and tear of punches. and they could control it once they replace the punch set. This is a cost which company has to bear. So once they did it, there was considerable control on variation, both with respect to friability as well as thickness and therefore other related parameters. Now this slide depicts about coating. You can see that most of the batches are uniform except one batch which is going haywire. And even this one batch can totally change the scenario. What if FDA picks up only this batch? Your premises will be locked. And therefore, it was very important that they optimize the spray rate, inlet, outlet, air temperature, all the atomizing air pressure, etc., and validate that so that there is no variation in the spray rate, and therefore the coating thickness doesn't change. If coating thickness doesn't change, you will get same disintegration, and then you will get same dissolution and hence the bioavailability. Your end goal is bioavailability. Packaging. Can we do anything in packaging was a question which was put by the management to the team, the tech transfer team, because they thought packaging was taking a lot of time. And yes, they indicated that the role change time contributes to 25 to 34% of total stoppage time. When you are changing the role between the batches, that is taking time. So can they optimize on that time? Answer was yes. So instead of having, say, rolls which were 1,000 meters, if they put 3,000-meter rolls, because the scale of manufacturing has increased, they could increase the efficiency to up to 65%. Original was about 40%. So such changes is what industry demands. You know that time also is money. And therefore, they would look at all the procedures by which they could actually make money at the same time, keep the quality. And this is GSK's product. Obviously, quality cannot be compromised. So, what was their control strategy? They have installed a high capacity blender. So earlier was 300 kg, they shifted to 1000 kg. They reduced the waste reduction, both in compression and packaging, by 35%. They released 10, 20 to 30% of the manpower. Even if that can be done, this manpower can be utilized for other purposes. And that again is beneficial to the company. Rework reduction by 50 to 70%. So all in all, there was improved process capability due to improved Six Sigma level, because they understood which are those parameters which we need to really critically control in order to give a more standardized process. So this is lead to lean, Six Sigma, continuous improvement and control. I hope you understood the basic steps now of Sigma method of DMAC. Now, look at what is QBD. You will be surprised that nowadays we have, just like karate black belts, you have Sigma black belts. But what are they doing? Actually, they are also doing QBD. Parameters are almost the same. Here also you are defining quality target product profile. For generic products, we say QTPP, and for innovator product, we say TPP, target product profile, because you don't have a reference standard there. Term which was used here is measure, synonym of which is determine. So you are determining critical quality attributes. Here you are analyzing, here also you are analyzing. So what are you doing? You are doing risk analysis. Here it was termed as cause and effect. Here it is termed as risk analysis, wherein you are linking the CMAs and CPPs, that is critical material attributes and critical process parameters, to critical quality attributes. As a result of which you are moving further and using design of experiment-based experimentation. So when you use DOE, you are creating design space. So what are you doing in turn? Is you are optimizing your formula. Here also you are improving and optimizing. Net result here is control, and here it is termed as control strategy. Isn't it very clear that steps are the same? So you have now planned set of controls of the materials and the processes so that you can get the desired QTPP. And of course, go on working on it in a continuous manner. So what is QBD? It is nothing but a systematic approach to product and process design as well as development. What are the main purposes? Definitely, we are looking at a meaningful product quality attribute, because in a long run, what we want to do is ensure clinical efficacy and safety. That is the objective of each and every worker, each and every pharmacist for that matter, to deliver products which are safe, which are efficacious, and which are not compromised in quality in any way. To achieve enhanced product and process design and understanding, because it's a iterative, reiterative process. As you can see here, continuously you are improving, and therefore your understanding about that product and the process goes on increasing over a period of time. To implement suitable controls to reduce product variability and hence reduce the risk to product quality. Yes. Why are you coming up with control strategy? This is the reason. You want to reduce the risks. And to reduce the risk, you need to optimize. So this can help you in registration and manufacturing. It has become now a regulatory norm. But when it was not a norm, definitely if you present the data in a systematic manner, any, it could be DCGI or it could be state FDAs, they would welcome this move, because you are able to better explain to them why your product is better than 10 others in the market. And overall, it can enhance the capability to manage post approval changes. I've already told you, because you are getting that design space. And if you are working within that design space, you are going to not go back to the regulatory agencies. So QBD is a process. First step is defining QTPP and TPP. Once you have listed down these, you are looking at critical quality attributes. You are listing all the material attributes which may be important. And you're also looking at process attributes which are very critical. In risk analysis metrics, you are linking these both. So you are just putting them as which of these parameter is high risk to achieve a quality attribute, which is the one which will have medium risk and which is the one which will have the low risk. So obviously low risk parameter you need not work on. Medium risk parameters with small amount of experimentation you can cater to, but it is the high risk parameters amongst these which you need to pay attention and see that you convert it to low risk by using DOE. And for this DOE, you typically nowadays people are taking help of softwares. In our days, we used to do it, of course, manually. And then arrive at a control strategy. All this will be very clear to you once you see the examples. But to explain the students in simple words, I tell them that you have to give exam and get minimum 80% passing. So can you list what are the CQAs, CMAs, CPPs, risk analysis, systemic experiments? What would you do? Many people say that if you have given that minimum 80% is passing, I will read enough and get 80%. But there are very few who will say that though 80% is the target, I'll work for 100% to get 80. So those who say 100%, that goes in TPP. Those who say 80%, close to 80%, there is a reference standard because I have given that problem. So that goes in QTPP. So what is desired? to get that particular marks, what is desired? Obviously, you need to have good books. You need to read through good journals talking about master students and PhD students. They should not be only limited to PowerPoint slides, which typically happens in case of most of the institutes. So you have to ask questions also where they can apply mind. So 80% is a passing criteria doesn't mean that you give 80 multiple choice questions and your work is done, or give one or two liners which are there in your PowerPoint and work is done. You need to make sure that they have understood whatever you have taught. For that, they'll need many primary sources of information. So they will list that down as CQA. So material obviously would be all these notes, the guidelines which are available, ICH guidelines, all the primary sources of information, etc. And the process which they are using. Some people, are fast learners, some are slow learners. Process for each one is different. Some people, they do not open the books even till the last moment, but they are intellectuals, so they can grasp whatever teacher is trying to teach them. So the process to achieve this QTPP or TPP may be different. So then they need to look at and do the SWOT analysis. Each student knows what are their weaknesses, believe me. They know what are their weaknesses. So it is our duty as teachers to tell them how will you look at your weaknesses and convert those weaknesses into strength. So typically, if there are 10 chapters for the final exam, every time you open the book, you are starting from first chapter. That is not what is desired. Read first chapter, read 10th chapter, then second, then eighth, so that from both sides you are really equally well versed. Nothing should go in option. So these are some of the quality characteristics which you have to build into people. So obviously they would do this risk analysis, and whether they are failing with respect to the time which they are giving, or whether they are failing with respect to the notes which they have with them, is what they need to pinpoint. And once they pinpoint, obviously then you can do their hand holding and tell them how to improve. There are remedial classes taken to tell them how to improve, so that that risk changes to low risk, as a result of which you get 100% passing percentage. Once you explain them this, I'm sure they'll be able to do any kind of DOE. Moving further, some of the basic terms very quickly I'll be taking. I'm not sure as to how many are from uh, our pharmaceutics field, but definitely remember that this can be applied all across. Pharmaceutical analysts can apply this, chemists need to apply this, ecology people obviously have this as an integral part, and so do pharmaceutics people. So DOE, pharmaceutical optimization, is nothing but application of statistical technique to get as perfect, as effective, and as functional a pharmaceutical product as is desired. Earlier, it used to be empirical, that is one factor at a time or one variable at a time. But now, over a period, it has changed to computer assisted statistical design, because you do have softwares available. So, advantages are that yes, you need to do fewer experiments and get to know major data. Now, what was happening in trial and error is that you could not predict interaction between the variables. You were fixing one parameter and then looking at other parameters, moving on to second and the third and the next. But here it is possible to estimate interactions which are happening. For example, again, if I'm making a sublingual tablet, you know that sublingual tablet is desired to give a very fast onset of action. So for fast onset of action, what would be put in the formulation is super disintegrants. Super disintegrant is one thing, but you're making a tablet, so you're putting binders also. So can you look at effect of binder and super disintegrant as two variables together in this approach? Trial and error approach, no. In case of this, it is possible. So that's the reason why people have shifted from earlier approaches to this. What are variables? Variables are the parameters. It could be process related, could be product related, which are going to affect the quality of finished product. You can divide them as independent variables and dependent variables. I also put them as, just so that students remember, these are input and these are output. So input are independent, output are dependent. So these again could be qualitative or quantitative. You are making an emulsion, whether you're using oil in water type of emulsifier or water in oil type of emulsifier, that is qualitative. You're using single rotary, double rotary, that is qualitative. But when we talk about concentration, what is a concentration of, say, tween 80, which is used as an emulsifier? Or what is a concentration of disintegrant? Or, for example, we discussed one sublingual tablet, concentration of binder, that becomes quantitative. So these are under control of formulation scientist. Those of you who are doing chemistry, you know that rate of reaction also depends on concentrations of reactants or maybe the catalyst which you have used. So they are the quantitative ones. So in case of chemistry people, yield will be the output factor. If it is chiral compound, chirality is the output factor. But in case of pharmaceutics, since I deal with pharmaceutics majorly, I'll give you those examples. Dependent variables are not under our control. But yes, if you can control the input factors, you can definitely get the desired output. So this output could be, for example, if you're making a tablet which is fast release sublingual, again let's take that example, you need faster onset of action. So that means it should disintegrate. Pharmacopoeial limit may be higher, but I would put it as 30 seconds because it is life saving. For example, nitroglycerin tablets or isosorbide dinitrate tablets are sublingual tablets. So if they do not release fast, how do you think that absorption would happen and the patient would be saved? So in that case, drug release is equally important. But if you are talking about, say, certain nano formulations, which are nowadays, very prevalent, particle size is important because finally it is a particle size which will be related to drug release. So this is about variables. Input variable which influence formulation characteristics are termed as factors. So as I said, independent variables therefore would be input factors and dependent will be output factors. Levels, value which is assigned to each factor is termed as level. Now, for example, if you are using tween 80 as an emulsifier, we know that tween 80 can be used at 5 to 20% weight by volume as an emulsifier. So you can design trials with 5, 10, and 20. So 5 in this case becomes a lower level, 10 becomes a medium level, and 20 becomes a higher level. So 5, 10, 20 is termed as level. Now these levels, because computers should understand, are changed to coding. So you are coding it as +1, 0, -1, or 0, 1, 2. So these are coded. Levels are coded. This is very important, and you should understand that change in response is obtained by change in input variables. When you have a linear effect, that means the effect changes linearly as the input variable changes. For example, if you're making a sustained release tablet, increasing the concentration of polymer will decrease the release rate. If it happens linearly, it is known as orthogonality. Take a conventional tablet, increasing the disintegrant is going to increase release. It is possible. Same way, increasing the binder concentration is going to increase hardness. All those are linear effects, so they come under orthogonality. Now, these effects could also be non-linear. Non-linear obviously represent lack of orthogonality. These can be divided as interaction or as confounding. Interaction ones can be explained. Confounding, the other name for confounding is confusing. That means those effects which are not linear and those which are not happening due to interaction are coming in confounding. In these cases, you need to relook at your data. Maybe you have not selected the right kind of input factors. Maybe your design is not good. So go back, work on it, and see that either you get linear or you get non-linear, because obviously at the end of any exercise, you cannot be more confused. You are doing this so that your work becomes easier. So you can see here what I have represented is that as the polymer concentration increases, you are getting the release effect. So release, obviously, if I plot say release here, release is decreasing when I go to high polymer level. And so this line slope is the same as this line slope. Whereas this indicates that the other ingredients which are there in the tablet are not interacting. Whereas if you see here, for another drug, this line slope, which is on the upper side, is different than the lower side line. That means besides polymer, there is something else which is interacting, because of which there is a change. So this is interaction mode. So generally by looking at the data, we get most of the information, but definitely tools become handy when there's too much of data. Now, as far as our experiments are concerned, part of space which you are experimenting with is the experimental domain. Optimal region is a design space which you get by mapping of the responses. And then you have a control strategy, which if you use every time, you will be within the design space. The methodologies which are used include simultaneous optimization method and sequential optimization method. Please keep in mind there are n number of these methodologies, but industry is majorly using, if it is possible, if you have less number of factors, go for full factorial design. If the number of factors is more, industry is using either box Bentham or central composite design, and you'll know why. Definitely in cases where there are 2 or 3 ingredients, maximum 4, and their addition effect is 100%, mixture design and de-optimal design are also being used. Very few cases. Like for example, those of you who are working in SMEDs and microemulsions would know that these designs will be much more useful than the central composite and box Bentham, because you're plotting ternary diagrams. Plackett-Berman is typically used as a screening design because it uses number of levels more, but operates only at less number of levels. Next 2 slides are most important, and I will pass on this presentation also so that you would know which design to use what. No one has done this exercise except us, and it is there in our book also, which we have published along with many case studies. So factorial design operates only when factors are less than or equal to 3, works at 2 or 3 levels. Fractional factorial, when factors are more than 3, rather more than 4, works only at 2 levels. Plackett-Berman, as I told you, number of factors increases you are using this, but number of levels at which you can use is only 2. So this is majorly used as a screening design. Mixture design also used when factors are more, and it also works at more number of levels. But in this case, interaction of factor at each level cannot be estimated. Again, this is used majorly for preliminary screening. D-optimal is also a little complex model, but pay attention to central composite and box Benchem. Both of these operate when factors are less than 6. In central composite design, levels are 5. So that means number of experiments which you will do in case of CCD are more than number of experiments which you will do in case of BBD. Both of them are quadratic model. That means it will give you idea about interaction. So it's very important that you understand these. There are no negative points, I would say, of both the methodologies. Of course, if you have many variables, fractions, it is difficult to predict, but nowadays softwares do that job also in a very easy manner. So what are stages of optimization? You are first selecting influential variables or input variables. You are assigning values to those influential variables. Then you are selecting the experimental designs from whatever we discussed in last 2 slides, depending on what levels you have fixed. Then you are carrying out the formulations. Then you are doing the statistical evaluation, and finally the validation. This is also how experimental designs actually can be classified. You can classify them on basis of which model and which application. So model could be linear or non-linear. So non-linear, I told you, quadratic models, you have CCD and BBD, whereas linear models are fractional design, factorial fractional design, and Plackett-Bowman. Application wise, for screening you can use some, for optimization you can use some. So this is just another way of putting the things which you should know. And I'm sure Sharwar would have taken this in detail, but these are the softwares which are typically used, of which, to my experience, I have used most of them, but Design Expert and Minitab are really very easy and user friendly. So we have both these, and we also have Jump. Now I'll take a case study so that things are easy to explain to you. This is the in-house study which we have done. The product has gone, actually been technology transfer has happened. So some of the ingredients I'll mask, but you'll get a very fair idea about how one does work. So this is about generic development of adsorbed aprepitant nanosuspension. Now, aprepitant is ingredient which is used for chemotherapy-induced nausea and vomiting. In market, it is available as Emend. Route of administration, of course, we wanted to keep it same as a marketed formulation, Emend. So we've also kept it as oral. So first step is we actually look at patent. And if you look at patent, this particular patent has been assigned to Merck Shop and Dom. The date 2012, and you know that 20 years it has life. So patent is still not expired. So you need to, that means if you want to come in market, you have to come out with a strategy which is not infringing on this. I'm sure all of you would have heard of para 4 filing. Para 4 filing is when you submit your application to US FDA saying that I am not infringing on the technology which the innovator has given, or you invalidate their patent, one of these. So in our case, what we wanted to develop is, and we do that very often, we develop technologies and keep by para 4. So whichever industry approaches us, they are free to take it. If they want to wait and file by para 3, that also they can do. Para 3 is they enter into the market only after the patent has expired. So either ways, we are in benefit because we have developed the technology, we have the know-how. It's our duty to see whether we give all exclusive rights or we give non-exclusive. So idea is that ICT also gets lot of royalty because we are deemed university and we need to generate our own funds in a long run. So when we looked at this particular landscape of EMENT, we saw that they had used media mill. If you look at the orange book, the patent was going to expire in 2027. And you can see that still there is time. Stabilizers which they have used for this technology were SLS and HPC. And their technology was that they will load this carrier drug, which is nanonized, onto sucrose beads. So there's a coating step. And believe me, this work has been done by master's student. I told him to write the QTPP, and QTPP would be to, as far as possible, because it's a generic product, only we are doing it by para 4, as far as possible, keep the target same. So if dosage form in the market is capsule, we would also want to go with capsule, not with tablet. It has to be immediate release because we need faster onset of action. This is for nausea, vomiting. So obviously, and that also after chemotherapy. So immediately drug should be available. There were 2 doses in which it was available. We said we'll do the same because that is what is innovator doing. Oral was a recommended route. This is something which people miss, and therefore I'm pinpointing here. And even, when you are teaching quality control to all the students, that parenteral product has to be meeting with sterility test, but all other products, including oral and topical, have to meet microbiological standards. And in finished product QC, I hardly see students writing, because we get MPharm students from, all over India. Most of them do not write microbial load. So that is absolutely incorrect. So obviously it has to meet pharmacopoeial standards, content uniformity, in vitro release, etc. Everything as far as possible has to be same to innovator. Marketed was providing it as a strip pack. So obviously first you will go with strip pack. Now please know that blister pack is cheaper. First you introduce, because marketed people, innovator has put it in strip, meaning they have checked the compatibility with strip. So you are gaining time there. So first thing is put it in the market with strip. Later on, you can do studies, stability studies have to be done for any product. So you can simultaneously do and see whether your blister pack can also serve the purpose and protect the drug. If it does, go with blister. Who stops you? And then generate the stability data. This is what, again, my students have to do before they start work. They have to convince me on paper what technology they will use. And I ask at this stage 100 questions. So to understand what is in their mind, what hypothesis they want to go ahead, do their handholding wherever it is required. So the student suggested that we will not be using media mill, but we will use HPH, which we also have. We have both. We have planetary ball mill, and we do also have HPH as well as microfluidizer. So he said, we will use HPH. Now, HPH, which is the lab scale, has a limitation that it can take particle size of a certain range. Otherwise, it gets clogged. So obviously, if you know the material characteristics, the drug characteristics, either get the drug of that size or change it to that size by milling and then process. So this is what was proposed, that we take drug, wetting agent, stabilizer, and water, do ultra-terex milling followed by HPH to get a liquid nanosuspension, and then take this liquid nanosuspension and absorb it onto an inert carrier to get a powder which is filled in capsules. Now, innovator has filled beads in capsule. We are saying we'll fill powder in capsules. Is it acceptable? Answer is yes, because final dosage form is capsule. So all your quality characteristics should match. We are also giving capsule. What I told at this stage is to student is, see if you can get out of their patent even with respect to the wetting and stabilizing agents which they have given. And if you remember, they have used SLS and HPC. So we took up that challenge and moved ahead. So then we wrote critical quality attributes, whatever are applicable to the nanosystems and to the capsules need to be listed. Of which then you need to write, first list down all, then write whether it is a CQA, whether it's a critical parameter. So in our case, yes, pre-milled particle size was a critical one because otherwise our HPH would not work. Final nanosuspension particle size was also important because that will govern the final solubility as well as bioavailability. Release is critical because we want to be same as far as possible, qualitatively same, right? So it has to be similar to market it, because if this is similar, we will get the pharmacokinetic parameters similar. But this doesn't always happen. And that's the reason why that industry came to us, because they had developed a dosage form, but it was passing drug release, but it was failing in bio. Whereas a product which we have developed, you'll see how does it perform. So then we made risk assessment matrix. We looked at CMAs and CPPs and related it to the CQAs, which were particle size, drug content, and rate and extent of release. I told you the low ones you need not pay attention, because if you take care of the parameters, the drug was very robust. So not that generated a lot of impurity. So there was no problem with respect to content, etc. But yes, we need to look at how these parameters can affect, and homogenization pressure and cycle both are known to affect particle size as well as the release. That they are directly related. So how did we do the study? It is a systematic study. I told the student, let's evaluate what those people have given, that means SLS and HPC, but let us also evaluate n number of other polymers which they have used as stabilizers and wetting agent. So suggested that contact angle study could be done. And you can see here, this is the data of contact angle with SLS, and this is the data with stabilizer 1. I have coded it because this has gone to the industry. I cannot reveal it because of the CDA. But gives you idea that we've done contact angle study, and we've also done saturation solubility study. Now you will see that SLS has definitely given them good wetting, but the drug is so soluble in SLS. That means, and they have used this as a wetting agent. That means over a period of time, there are all the chances that whatever nanoparticles you have made slowly go into this surfactant. And that can change the release behavior and therefore can change the bioavailability too. So we wanted stabilizers which drug is unstable because it's a suspension, right? You want a suspension. So HPC did perform well here, but we had 2-3 stabilizers in which drug was not soluble. So we don't want drug soluble in these stabilizer. At the same time, we want that it should be able to wet these systems. So we selected stabilizers 1 and 2. By empirical approach, we optimize the CMAs, wherein we have shown that if we use stabilizer 1 and 2 in combination, and that also 1:1 combination, particle size remains stable over a period of time. Why did we look at 24 hours? Because at least, once a batch is manufactured, to adsorb it onto an adsorbent, maybe 1 or 2 shifts will go. So keeping that in mind, we wanted that at least for 24 hours, this nanosuspension should be stable. Of course, this nanosuspension was stable for 7 days, but it was important that we do it for a longer period of time. So we went ahead, we did pre-milling at different times, and you can see that as we are increasing the time, the particle size is going on decreasing. And definitely we got the desired particle size of pre-milling at 5 minutes. So we went ahead with that. Next was HPH. So for HPH, we designated 3 levels, both for homogenization pressure as well as homogenization cycles. And the parameter which was our response or the output factor was particle size. Very, very important parameter. So now what we have used is 3 raised to 2 full factorial design, because there are only 2 variables. These are 2 variables and we are using them at 3 levels. This is another mistake people do. They write 2 is to 3. Please remember, this is indicating input factor and this is indicating levels. So when we use 3 is to 2 full factorial design and started with design expert, of course we could have done it manually, but since it was available, all you have to give is what I had given in the last slide. What levels you will use of both the parameters. So then design expert will give you this design wherein they'll say which parameter to be used in which run and what is a response which you expect. So this is a response which software has given you. Then you do all these runs. 3 is to 2 means 3 into 3, that is 9. So run 9 experiments and get the results. Now, next step is that design expert will ask you whether you want to apply for any transformation. If you are not sure, you can write none. But here we knew that as we had taken some preliminary runs, so we knew that as we are increasing the number of cycles or as we are increasing the pressure, the particle size is decreasing. So we have put inverse transformation. But if you don't know, put none, automatically software will take you to inverse. We move ahead, we get such a chart. Now we need to understand this chart. And in fact, again, software helps you. It analyzes the data and tells you whether the model which you are using is correct or incorrect. So you can see here that we have got F value as 21.53, which is a very high value. And this probability value, P value, if it is less than 0.05, it does indicate that that model is the correct one. So in case of linear, you can see the value is very high and the value of probability is also less than 0.05. So that suggests that model value of 21.53 implies that the model is significant. And there's only 0.1% chance that F value this high can actually happen due to error. So it was very easy for us. Then we, of course, plotted different kind of curves. It could be 3D response curve, wherein you can see that as pressure is increasing, size is decreasing. Again, as number of cycles are increasing, size is almost remaining the same. But more than these contour plots and 3D response surface curves, I tell my students to look at this equation. This equation itself gives them information. You have factors x1, x2. X1 is pressure, x2 is number of cycles. Look at the sign which is in front of these factors. If the sign is negative, means they are inversely proportional. If sign is positive, that means they are directly proportional. So very clear that as we increase pressure, size decreases. And as we increase number of cycles, also size decreases. Once you do that, you need to actually validate. So pick up any of the points from the design space. You do get design space. From design space, you can check. In fact, when you use design expert, you'll know that as you move your cursor, value will go on changing. So you know which particle size you want, right? So go on moving the cursor, and wherever you are getting that particular size, take that trial and do that thrice. So we got to know that, yes, I can process at 1200 bars and use 30 cycles to get whatever size we desire. And we wanted submicron size. What was given by software was 836, and what we got after validation was 805. So plus minus 30 nanometer, generally you do get variation, and that is acceptable variation. But the correlation of experimental value with the model predicted value was within 95% confidence level. So what have we done? Now we have updated the risk analysis. That means wherever you had seen red, yellow, etc. earlier, all has changed to low. And that also we have gone a systematic way. So next step was, can we avoid coating? So can we actually look at whether we can absorb this? And answer was yes. So this was a second step. For that, again, we wrote down the CQAs. And here you know that you're converting into powder. So you want powder to be free flowing in order to be put into capsules. So it becomes very important for us to control the flow behavior, it becomes very important for us to control moisture so that you don't have a sticky powder. So obviously, if you control these, all other parameters would be controlled. At the same time, by adsorbing, we don't want to change the particle size. But we had earlier data on this with many other drugs. So we knew that it doesn't have much bearing. So we had to, in a nutshell, control these parameters. We tried various adsorbents, but again, we know that aeropearl performs well. Again, like 1995 onwards, I've been working on these systems. We know for which systems what to use. So aeropearl 300, 12 ml it could take, 1 gram could take 12 ml of nanosuspension. That was sufficient. So we looked at flow properties, they were excellent. So we filled it into capsules. Again, the results of these were as per pharmacopoeia acceptable. We did this in vitro dissolution testing, and you can see that it matched entirely with marketed. And at this stage, our palpitations started. Why palpitations started? Because the industry which came to us said that we are also getting similar profile, but we are failing in bio. But I was sure of the stabilizers and wetting agents which we had selected. So we went ahead and did in vivo studies. And you can see that this is a marketed dosage form, and this is what we have prepared. This is by coating, this is by media milling, lot of variations we are getting. Whereas in our system, there are no variations. And plus, our kind of systems, we know that are able to really cater to effect of food also. So we were within 85 to 120%, relative bioavailability of 92.71%, which was very, very good. So we could confirm bioequivalence. Why? Only because we went in a systematic manner, applying QBD principles, understanding pharmaceutics and understanding even physical pharmacy well, to take care and develop this particular dosage form, which is now, of course, given to industry. So this was the updated risk, and this is the overall control strategy now. So we have used two stabilizers. We have used drug to surfactant ratio 1:1. We have used ultrateric speed 5 minutes. We have used 1200 bars and 30 as a homogenization cycle number. As far as solid nanosuspension is concerned, we have used this adsorbent, aeropearl. Bulk density is 0.3, so easily it can be filled in capsules. Moisture content also is under control. So in a nutshell, we are saying that this particular design pattern is allowing us to control all the parameters to the level of 6 sigma. Because after we finish, in institute, we can do only 1 kg, 2 kg. But this has been now scaled up to 1000 kg batch, and there are no issues. It just indicates that we've gone the right way. I will take another case study wherein we have used PAT tools, and this study was done with the University of Bradford. So I thought it will be important for me to put this. In this case, we have developed suppositories, which are self-microemulsifying suppositories of an anti-malarial drug, which is beta-artemether. Idea is, beta-artemether is a WHO listed drug. Idea is, in places like Africa, etc., they do not have primary health centers everywhere. And if they get infected with malarial parasite, most of the kids die. There is one death happening every 5 seconds, which is a very, very different kind of a statistics. So I couldn't take it. So we said, by the time these people reach hospital, can we give something to them at home which can arrest the growth of malarial parasite? And with that in mind, we developed self-microemulsifying suppository. Why self-microemulsifying? Because we want the action to be fast. So there are suppositories in market, but they are no good. So this particular drug belongs to BCS class 2. So there are dissolution controlled challenges. So therefore we said we'll make self-microemulsifying systems and make suppositories which are based on SMEs. Advantages will be there. Of course, there'll be faster softening because they are SMEs based. Rectal route, so it will bypass first pass metabolism, so higher absorption. Immediately it will microemulsify in the rectal cavity and therefore lead to better absorption all over the rectal mucosa. Obviously, because we have converted into self microemulsifiable systems, drug will be soluble as against the present suppositories, and therefore it will give a higher absorption. First time in the world kind of product. So TPP, we had to list, we didn't have any reference standard. So we listed all the target product profile. To start with, we went ahead with the same dose at which marketed suppositories are available. Of course, marketed are not SMEs suppositories. So everything you have to list down, know what liquefaction time you expect, what mechanical strength it should have, because suppositories should not melt in the packaging material. That was another challenge which I had given to this student. This student was also a master's student, and in fact a BTech student, undergraduate student, who worked on this, and then we took this work further with help of master's student. So what the challenge was given, can we come up with a base which is room temperature stable so that these suppositories need not be stored in refrigerator? You know that most of the suppositories which are available, labeling says store in refrigerator because otherwise they melt. So challenge was come up with a base which is stable. So this was a schematic again, paper schematic. And therefore, for me, it becomes very easy to take such lectures because hypothesis always is in place. What was proposed? We would use melt mixing as a technique. We will add microemulsion pre-concentrate, that is self-microemulsifying pre-concentrate, to this melt mix. Once that is done, we will do the suppository moldings. But now what has come in, which is new here? We are saying that these suppositories are not a batch process. This can be done on a continuous mode. And how it can be done on a continuous mode? That is, make sure that you have blend uniformity. If there's no blend uniformity, it goes back to the container. If there is uniform blend, it goes ahead, gets mixed with the microemulsion pre-concentrate. And this particular pre-concentrate, you are checking again for uniform mixing using NIR probes. If it is good, it goes ahead. If not good, goes again back. One needs to, of course, control the temperature, which is very important because suppository base should not be overheated. Once this is done, it goes to the mold suppository molding machine, which is a continuous suppository molding machine. And then from there on, it will be going for packaging. So there are in-process testings, which I have mentioned here at each stage. For some kind of suppositories, we have done work on control release suppositories also. For some, you can have viscosity as a online parameter too. But here it was not necessary because we were aiming at fast release suppositories. So then we listed the CQAs, and you can see the CQAs here, the blend assay, the self microemulsification time, the droplet size which you get after it comes in contact with rectal fluids, and the polydispersity index. All of these were yes. And then we had to actually convert them really in a very systematic manner, looking at what is available in the literature and what we really need to work. And can see a lot of reds here as against the earlier case study. So we did CMA optimization. For this CMA optimization, what I did is we looked at different supposiers which are available. Supposiers are available from Gatafos, which is a France-based company. What was important for us is if we are going to incorporate our self-microemulsifying system in a base, finally it should be dispersible. If base does not release the SME, how will the drug get absorbed? So you can see that in all other bases it was insoluble, but in supposier AP it was dispersible. So these are the studies which are very important and are warranted to be done. In fact, if you look at pharmacopoeias, use of hard fat is recommended for rectal and vaginal use. And this particular grade of supposier AP is a water dispersible grade, which also comes from a hard fat. And we have used artemether along with these ingredients because it has given very good solubility in these ingredients. Some of these ingredients, like transcutol P, are also known to be permeation enhancers. So that also further gives justification, enough justification to say that we'll get faster onset of action. So this is a tentative formulation composition. I've given the ranges, not the exact quantities. The target, of course, is that it should give self-microemulsification in 45 seconds, and the droplet size should be as less as possible, as less than 150 nanometer is what we had proposed. So in this case, we went with triplots. And you can see that why we could go, because there were only few ingredients. So when you have few ingredients, you can plot these, and softwares for plotting these are also available. And you can, again, just like earlier case I had told you, if you move the cursor, from one place to another, you'll know in which region you'll get 150 nanometer size. Very clearly it can be seen, it's written here. So go to that point, read the ternary plot, what would be the ingredients which could be used, and use those ingredients. So when we did the overlay plot, this is a huge design space which we got. So variety of ranges were investigated, but design space we could limit to 8 to 12% of supposier, and so on and so forth. So once we got that, of course, the next step would be melt mixing. We looked at inline NIR probes to see whether there was uniform blending or not. So when we realized that it requires at least 15 minutes of mixing, it went ahead. Not only mixing, rate of cooling is also very important, because that suppositories need to set. If it is absolutely fast, in that case, you can get the drug crystallizing out. At times, it can cause that. So self-microemulsifying in this case was beyond specifications. So we thought that we will optimize a rate of cooling as 1 to 3 degrees Celsius per minute. So we have updated the risk metrics. You can see wherever I have used PAT techniques, I have indicated. So this became now the overall control strategy, wherein we have used 40 milligram of drug per suppository, supposier along with gelisar and transcutol. And again, this technology is also being explored by pharmaceutical industry right now. We are hopeful that even this will be commercialized. So I would like to say that by giving these two examples, there is enough evidence that if you develop your products using quality by design approach, you can achieve 6 Sigma. So QBD is nothing but a 6 Sigma alignment strategy for pharma excellence. So my earnest request to all those who are attending to know your formulation very well, to know your critical materials very well, to identify the critical steps very well. So typically when I take my master's lectures, even my B.Pharm lectures, we tell them to write a flow chart of any particular process. And typically, I take it for a liquid suspension. Solutions are easier. Then I take it for semi-solid of one each type. It could be ointment base, which is oleaginous. It could be cream because it requires 2 phases. It could be gel and emulgel, which is nowadays becoming very popular. Then I move to aerosols, and I take for tablets by direct compression and by wet granulation. And there I give them challenges like the one which we today discussed. It could be sublingual tablets, or it could be oral mouth dissolving films. It could also be buccal tablet control release. Move on to Oros tablets, osmotic tablets. I give them examples even of parenteral. So all these are done as group exercises in the class. Some are given as individual until they understand the aspects well, and then some are given as group exercises wherein these groups need to come together, brainstorm, and present to full class. And my modus operandi is one group presents. Our classes are small. We have only 30 students. So the other groups mark these classes and I also mark them. And you'll be surprised that most of the times my marking matches with their marking. Total unbiased marking. Because they also know that at end of the semester, all of them will be actually so very well trained that they will get good grades, which they do. Meaning, initially, they get 1 on 5, 1.5 on 5, and by the time they come to parenteral dosage forms, even including freeze-dried, lyophilized, to be reconstituted products, they come to 5 on 5. So that's a kind of achievement, which we get if we inculcate the right practices in them. And in doing so, they need to start with suggesting a prototype formula. There are marks at each stage. So if I give them to make antacid tablets, they need to propose which API they will use, which formula they will use. So obviously, they'll go ahead and read about the API characteristics. If they read about the API characteristics, they know whether the drug has polymorphism issues, whether the drug is low melting, whether it is pH stable, temperature stable. Accordingly, material attributes they can pinpoint. Same way, they need to look at process. So when they make the flow chart, I tell them write the flow chart. On one side, write the in-process QC. On other side, write which parameters you will control. So each stage, first, when the group presents, the whole class deliberates. And we make the very holistic picture of the flow chart. And then we move to QBD. We give them temporary access to design expert. They are able to do their own design. Those students who are really keen, believe me, they have done at B.Pharm level better work than PhDs in many universities. So they come to lab, they do work, they publish also along with us. So what all in all, what I'm trying to say is that teach them the right way. And this is a book which I was talking to you about on pharmaceutical product development. It's there in PCI syllabus too. And we have given variety of case studies. And I've been able to do this book along with, of course, Dr. John D'Souza and Maruk Rustamji. Now, Dr. Maruk Rustamji has about 35 years of industrial experience. And my association also with industry has really helped in shaping this book. So we have given many case studies which you can pick up and give it to your students. You can change the name of drugs and then they can do a definitely better job. And of course, another book which is on nano because we are in that area. And with that, I would like to thank once again the organizers to give me this opportunity to share Six Sigma as well as a QBD technique. I have purposely finished it before time so that I can answer all questions, queries which you might have. And I'll fail in my duty if I do not thank my research group, which is a very dynamic group, as you can see, ever smiling group, adding to all what we are doing on a daily basis. And of course, my alma mater as well as my Karma Bhumi Institute of Chemical Technology, which gives us reasons to be happy every day. Thank you so much.
Wow, ma'am, I must say it was excellent session. Like last time, much in fact better, and going better. And I wish I could also have this opportunity to work under you. Maybe I get it someday. I really hope that.
All are welcome. Just let the COVID pass. I'm sure we can all, whatever stage of life you are, if you need help, I'm always available.
Thank you so much, ma'am. That is so nice of you, ma'am. We have questions which I would request the delegate to unmute and ask, ma'am. I will first request Udayveer Singh Sara. Sir, if you are there, please unmute and ask your question to ma'am.
Good afternoon, ma'am. It's very pleasant listening to you always, and at the end, the photographs of your research team and the photograph of your institute. I had penned down a lot of questions to Dr. Shilpa, ma'am, and ultimately during the course of your talk, all the questions were answered, so thank you very much. But just one thing, could you throw some light on RTRT, that is real-time release testing, and something about QBD design in powder blends?
QBD design and powder blends.
Yes.
So basically, always you should start with end goal in mind. So if this particular powder you want to fill into capsules and sell, or this powder will be in pouches and then would be sold. So design actually will remain the same, I would say, since it is a powder dosage form. Majorly, it will be the. Could you be a little more specific, sir? Because if it is a powder, a drug powder, which is in
It's a powder blend, suppose.
but powder blend to be filled into pouches to be just like electoral powder, or just like the case study which I have taken where I filled powder into capsules.
I think it would be better if you take the example of the pouch, like electrol type.
Okay, so if you take that, again, what is very important is you write down the QTPP because you know what does electrol contain. So it contains a variety of salts. So major challenge there is that all of these salts are hygroscopic. So that means the moisture content is one very, very critical parameter there. So along with flow properties, I will give lot of emphasis on moisture content. And these powder blends, which are like for electral, are made using spray drying as a technique. So that means you need to have process parameters in place. And just like I mentioned in case of, I'd taken ranitidine case study, wherein I said that inlet and outlet pressure, as well as the solid content which is there in the amount which is getting sprayed, is playing an important role. They need to be controlled. Plus, once spray drying is done at the packaging level, these pouches, humidity of the area is equally important. So that's another critical parameter which needs to be controlled. So I would say majorly it would be the flow properties. Second will be the moisture content. And of course, the third would be all the critical process parameter wise, it would be the spray essentials. I hope I have answered.
Yeah, ma'am, briefly you could answer my question. I got to brief you about this because I've been teaching M-Pharm pharmaceutics and I've been using the NASH textbook as well as some other textbooks. But it was nice to see your textbook, and definitely I will purchase it and try to get more knowledge. Thank you so much, Dr. Vandana. It was very nice interacting with you.
Thank you so much.
Thank
Bye.
you so much. Sir, another question is from Dr. Anoop. Anoop, sir, if you can unmute yourself. Yes.
Yeah, hi ma'am. Very good afternoon, ma'am. Very nice presentations it was. Actually, you have already answered my query. My query was related to ranitidine containing products. My query was, is manufacturer not followed the QBD in case of ranitidine, and how these genotoxic impurities crosses the limit, particularly in ranitidine containing products?
So thanks for that question. So this particular case study, which is available no by Six Sigma, is a little older study. I would say 2016, when QBD was not a regulatory requirement. Now it has become a regulatory requirement. So, but to answer your question a little different way, many times it is not that impurities are not present. It is only that the techniques which can detect those are not present. So, as the instrumental techniques are also advancing, right? So, nowadays, even by using computer aided design, you can actually know whether the compound will be xenotoxic or not. So it may be present at that stage also, but may not be detected because the quantifying techniques were not that robust at that particular time. So now that it has come to light that this genotoxic impurity is there, right? People have to actually test their products, and almost all the products which are there in the market have this particular impurity. Only thing is that what they are indicating is then there are certain food products which also have the same nitrosamine impurity. So if in food products you are taking, always in more amounts than these tablets, which are small tablets. So how relevant is this question to be asked? And my answer to that always is that anything in excess is a poison. I'm sure each drug, when it goes through a drug development pathway, what FDA is looking at is risk to benefit ratio. So if benefits are more, it comes in market. For that one small thing which could be genotoxic in amounts which are definitely more, and you're not giving, it all depends on how often people are popping up antacids. So matter is that whether such products should be made prescription products or they should be available OTC. That is a major issue which needs to be tackled, rather than whether impurity is there or not. All chemicals are no good. Let me put that in one sentence. Sorry for that, I'm also a pharmacist.
Thank you, ma'am. We have a question from Dr. Chitra. Chitra, ma'am, you also wanted to ask a question. You had unmuted yourself. Chitra, ma'am. I think it was my mistake. Oh, there is one question from Dr. Reddy, ma'am. Though you had already answered in your PPT, can you mention any free tools available for DOE?
So that way, in fact, for 3 month version is available even of your design expert. So you can download the earlier versions are always available free. And if you're interested, this company, JMP Jump, they are providing nowadays free to academia so that they can get hands-on experience and later on maybe buy. But they
Right.
are not very expensive. I think. You can go ahead with buying them if it is your institute buying the same. What we have done is in each of the lab we have these softwares, because you can talk to the dealers many times. You can say that I will publish say maybe 3 to 4 papers using your softwares in a year. They'll give you free because their name is getting basically getting popularized.
Right. Chitra, ma'am, I guess you want to ask a question. Chitra, ma'am, can you hear me? Ma'am has unmuted herself, but Chitra, ma'am? No, I guess. So thank you, ma'am, once again for this wonderful session. In anybody else wants to ask a question to ma'am, may please unmute and ask ma'am. Anybody else? No, ma'am, I guess nobody for
If
now.
not, I will give them exercise.
Yes. Yes, ma'am.
Yeah, so typically what we do is, as I mentioned, we give, say, drug A, which is a potent drug. Potent means it is a low dose drug, right? 1 milligram.
Yes, ma'am.
Now, this particular drug is light sensitive,
Okay.
belongs to BCS class 4,
Okay.
and I want to make a oral conventional tablet of this particular drug. Now let's hear from teachers what would be their prototype formula. I hope we have time, Dr.
Yes,
Shilpa.
yes, ma'am. We have 20 minutes. So,
So anyone who is ready will be raising the hand. I repeat, drug is a potent drug, dose 1 milligram. So, which is unstable in presence of light and is BCS class 4.
yes, anybody who knows the answer can raise their hand, please. Yes, yes, Chitra ma'am would like to answer. Chitra ma'am?
Chitra
Chitra
madam
ma'am
saying that
is
she's
not,
not able to unmute.
yeah,
Unable to unmute.
yes, she's unmuting but not audible. Chitra ma'am, if you can put it in the chat box, we are not able to hear you. Or maybe you can join some other system if okay. Chitra ma'am?
Yeah, now she can unmute.
She was doing it earlier also, but we were not able to hear her. Anybody else? If Chitra ma'am, we are not able to hear. Yes, Chitra ma'am. Ma'am, can you join some other system? Maybe your voice is not audible from your system.
She's
Am I
actually,
audible now?
Yes, yes, yes. We can
yeah.
hear you. Yes.
Okay. Hi, ma'am. It was a wonderful session and really very nicely explained.
Thank you
So
much.
I think the problem you have given us, can we approach it by adsorbing this drug on a suitable adsorbent and then giving coating to the drug? Because you said it is unstable and that coating has to be light protective coating. So is that the right approach?
One can do that. So again, meaning to tell you that there is no one right or wrong. That is other thing
Right.
which we evaluate with students. But you just said absorbing. But first
Yeah,
would
because
be,
you said it's potent. Now you said so. Even before adsorbing, we can go in for coating probably.
no. So thing is, you tackled one issue, but not tackled the other. It's BCS class 4. That means it's not soluble, it's not permeable. So even if you adsorb and fill it, say, in capsule or make a tablet and coat it, finally it is not going to get solubilized and not going to get released, isn't it? And
Right,
not
right,
permeating
right.
also. It is not permeating also.
Very
So what
true.
you need
I
to know, right, is BCS class 4
right,
is
right.
low solubility, low permeability.
Mhm.
And yes, you've tackled one part, okay, that is, it is unstable to light. So what can be done is coating. But what is more important again is that when you are dealing with such a drug, you know that if it is very highly light, unstable, you need to use sodium vapor lamps in the manufacturing area. So you cannot just make, say, tablet the way you make and then coat it. All this has to be done under sodium vapor lab. So normal light or normal UV light, nothing will be helpful. So industry approach would be to, just like for nifedipine, people use sodium vapor lamps. You would have seen the yellow lamps, right? So drug has to be processed under that. And that's the reason why I asked you this question. Other is, it is a very, very low dose, 1 milligram. So, and the minimum tablet, which you can make is 4 mm. Typically, in industry tablets, which you pick up, will be 4 mm or 8 mm. So, for 4 mm, that means 50 milligram. So, which are the ingredients which you can add, which can increase the solubility and permeability, and yet give you a tablet? So, I mean, it's not that you have to answer right now. I thought if you have something ready, you can.
Right, we need to think about it.
Madam, in
Definitely. So,
that case, we can make the prodrug of that drug to improve the permeability and
So-
enhance the solubility. Maybe an answer.
that is also one of the approach which can be used. So, but keep in mind, the moment you have made a pro-drug, meaning you are going back. And in what way you're going back? You have to do all the preclinical data. You have to do the clinical phase 1-2 data. That means you are again going to take 10 years to develop something, isn't it? So it's
Yes, thank you.
it's not a research problem only. Industry wants to move fast. So pro-drug, not that it cannot be done. I'm happy that you are thinking from that angle. If nothing works, this definitely can work. And again, the pro-drug has to be such that it gets metabolized and gives you the drug. What if the drug which I have given you, certain assumptions will be there. What if the drug which I have given you metabolizes in the body and gives you the right kind of drug?
Yes,
But doesn't matter, Anuj. I'm happy that you thought of this approach, and chemistry side, that could be one of the approach. Making different salt than what is existing could be another approach. Many times
yes,
just
yes,
changing the salt also can increase the solubility. But I, being from formulation background, I was looking at
ma'am,
it.
it can be given by the using an implant in the sustained release formulation design if it is for the certain period of the time. If we are giving a such drug,
Oral tablet, please.
oral tablet formulation. Okay, okay. Yeah.
Anyways, I think such are the challenges which you yourself should give to yourself and work on it. I'm not giving the answer now, Dr. Shilpa. Maybe next time, sometime, if I have a chance, let them.
Sure, ma'am. In
Teach
fact,
you
we can put this
yourself.
question. We have a WhatsApp group with us. We can hear out from them. Maybe they come to some approach which we can directly ask you later on this.
Sure. I mean, it's such a, I mean, if you can tackle such issues, most of the formulations you'll be able to make. And in fact, answer partly is there in my presentation itself. So you should have come up with at least that strategy, if not something else.
Right.
Yeah.
Right. Thank you so much, ma'am. Once again, it was a wonderful session. I'll just share my screen with everybody. Thank you, ma'am, once again, and please accept this certificate from our side, a small token of gesture from our side. Thank you, ma'am.
My pleasure always. And
And
even if there's no FDP, I am available to anyone, please.
that's so nice. We can even see you were attending many of our sessions in the FDP. That itself is a big, big honor for us.
I was keen to attend all, but unfortunately, afternoons I have my own lectures, so it
Right,
clashed. But
right.
nice to learn from everyone.
Thank you, ma'am. Thank you once again. Thanks a lot.
Bye bye.
Bye-bye, ma'am. Well, thank you, ma'am. Moving ahead, Kanika, ma'am, if you can see us, can you please unmute yourself and switch on your video?
Yeah, sure. Good afternoon to everyone.
Very
This
good
is Kanika.
afternoon. Yeah, good afternoon, ma'am. Nice to see you. And if with your permission, can we start the session?
Yeah, sure.
Thank you. So moving to our next technical session, we have with us Dr. Kanika Thakur. research scientist, modeling and simulation group at Certara Simcyp, Frankfurt, Germany. So, ma'am will be speaking on nanocarrier-based approaches for topical drug delivery, opportunities and challenges. So, welcome, ma'am, and I would like to give a brief introduction as well. So, ma'am, as I mentioned, is working as a research scientist in modeling and simulation group, Certara Simcyp, Frankfurt, Germany. She received her PhD from Punjab University, India, in the field of QBD oriented design and development of topical drug delivery systems. She has also been the recipient of Commonwealth Scholarship in 2017 and did 1 year of intensive research work at Center of Regenerative Medicine and Cellular Therapeutics, University of Nottingham, UK. Dr. Kanika has extensive experience in developing topical formulations, conducting in vitro and in vivo dermal absorption assessment studies. She has authored more than 25 publications and 5 book chapters in international journals in the domain of topical drug delivery for dermal diseases including psoriasis, atopic dermatitis, osteoarthritis, and burn wound infections. Ma'am has also presented her work in more than 15 conferences, both at the national and international level, across UK, USA, Europe, and Japan. So, welcome, ma'am, once again, and greetings from Lloyd Institute. And over to you.
Yeah, I would just share my screen.
Sure, ma'am.
Okay. Yeah. So first of all, thanks a lot, Dr. Shilpa, for this kind introduction. And I would also extend my gratitude to the organizers of this AICTE-sponsored FDP program on exploration of nascent pedagogical tools for updating teachers of pharmacy education, and also to the management of Lloyd Institute for having me here. It's always, good to have such kind of programs where you can exchange your ideas, your thoughts, as well as your research with other people. And I think the stage has already been set so good by respected Dr. Professor Vandana Patravale, ma'am. And I would say, ma'am, that I'm really a great fan of your research work. So whatever I'm presenting today, it's just a part of my PhD research work and a few other small research attempts which I did at University of Nottingham. So basically it carries and it covers most of the research areas which I have done in topical drug delivery. As you can see, the title of my presentation is Nanocarrier-based approaches for topical drug delivery and opportunities and challenges. So if we talk about the global share for topical drug market has increased to about like 6.4% from 2016 to 2021. And there has been an ever increasing demand for these topical products, mainly because of many reasons. First of all, there has been an increase in, I would say, topical diseases, skin infections, bacterial as well as fungal infections. And secondly, there has been a rise in antimicrobial resistance. Thirdly, the environmental factors, including pollution, etc., have been contributing a lot to these problems. So we see there are a number of opportunities which are there for the pharmaceutical companies to work in this area. So as we see, topical drug delivery might appear to be a very simple formulation strategy, just made up of some creams, suspensions, some solid as well as semi-solid formulations. However, if you go deep inside, the real story is somewhat different. There are a number of challenges which are associated with such kind of formulation of such kind of drug delivery systems. So as I said, that the market for topical drug is increasing, and the global health diseases burden report, which was just came out in 2020, it stated that topical diseases like psoriasis as well as atopic dermatitis, they are one of the top 4 reasons of how much burden they put on the healthcare system. And we haven't been able to come up with much of the solutions. The solutions which are available in the market, they are temporarily with recurrence of a number of such kind of diseases. And also, if we talk about the opportunities, a number of biologicals are being explored in order to deliver through this topical route, mainly because it is easier, and it is a kind of patient compliant way in which you can administer drugs to the patients. So I would be talking all about this, and also I would be talking about a bit about the potential of nanocarrier based approaches with a few examples from my own publications, and then I would be talking about the formulation software, this kind of new strategy which is there, that's formulating for efficacy, as well as the PBPK modeling and simulation, a bit of it, which is my current area of work and which I have been working on in the oral and the dermal side within Certara Simcyp. So as I said, that topical drug delivery, it seems to be very easy. It seems to be, the formulation development seems to be very, fascinating and compliant. However, the real story is not so. If we talk about the oral as well as the parenteral routes, they have been associated with a number of disadvantages. While if we say that oral route is mainly accompanied by the first effects of the first pass hepatic metabolism, as well as the poor bioavailability, which is mainly associated with the oral administration. If we talk about the parenteral routes, there are always chances of exposure to multiple organs. So where drug concentration might increase, and that might get a damage to the various organs which are present in the body. So advances in pharmaceutical technology have encouraged the scientists always to look for alternative routes of drug administration. And apart from oral and parenteral, we land up here in topical drug delivery. Now, topical drug delivery also does not apply to skin only. It may include the rectal routes of administration, nasal, ophthalmic, as well as the vaginal route of administration, in which the formulation is such to permeate across a membrane or permeate across a barrier. In this presentation, I would be specifically talking about okay, so I thought to just have the laser pointer. So in this presentation, I would be specifically talking about the skin administration as a route, and mainly focusing on psoriasis, atopic dermatitis, and the burn wound infections. Now, if we talk about the skin physiology, basically it is an organ which protects the body from microorganisms, and it contributes to about 4% to the body weight. However, the physiology of the skin is quite complex and quite intricate. As you can see, the topmost barrier to the skin, that is the stratum corneum, is the major barrier to the permeation of most of the molecules, and also the physicochemical properties of the drug molecules, including molecular weight, the pH, the pKa solubility, they play an important as well as decisive factor to decide as well as facilitate the permeation of the drug molecule across the skin barrier. Then coming upon to the second layer, which is the epidermis, followed by the dermis as well as the hypodermis. Now, in case of epidermis, is basically composed of complex cell layers consisting of stratum basal, in which you can see the cells are basically columnar in shape, stratum spinosum, where the cells are almost polygonal, followed by stratum granulosum and stratum lucidium. So stratum lucidium is mostly present in hands as well as feet. As it can be seen, the entire skin structure is not so uniform as it seems to be from outside. Basically, the stratum corneum is composed of keratin-containing cells and intercellular lipid as well as ceramides, while epidermis, on the other hand, in addition to keratin as well as ceramides, contains a large amount of cholesterol as well as fatty acids. So the first important barrier to the movement of the molecules across the skin layer is permeation through this barrier, which includes partitioning, and then it involves diffusion across these layers in order to reach the target site of action. Followed by epidermis, we have got dermis as well as hypodermis, and basically collagen is present in dermis, which helps to retain the structure of the skin. Because of this complex physiology of skin, obviously there are multiple varied routes by which drugs can penetrate across the skin barrier. Talking about most of the drugs, they take an intercellular route of movement through the drug barrier, through the skin barrier. So in this case, usually the molecules have to be less than 0.6 kilodalton in order to be able to penetrate through the skin. So molecules which are above 500, they cannot really they don't have the right physicochemical properties to penetrate through this route, because in this case, the drug molecule will penetrate through the distance which is present between these keratinocytes, involving the movement through intercellular lipid as well as ceramides. While if we talk about transcellular involves the movement of the drug molecule across the keratinocyte. So in this case also, the molecule has to make up a sort of balancing approach between hydrophilicity as well as lipophilicity to be able to move across the skin. Then we have transepidential route, which involves the movement through the hair follicles, and a number of water-soluble substances usually take this route in order to navigate their path through the skin. So the entire range of topical formulations which are available, they have to take considerations of the physiological factors which are associated with a particular kind of disease, and also the physicochemical properties of the drug molecule which is to be incorporated into the formulation for the delivery. And this optimum approach can then be used in order to deliver the drug at the desired site of action for the treatment of various skin diseases. Now, if we talk about various skin diseases, I just land up thinking that are there any permanent solutions which we might get? Because if you see the kind of skin diseases which are there, they always have certain kind of recurrence. You apply the formulation, it gets good, and maybe after 6-7 months, again, there is another recurrence of the same kind of disease. And we have a kind of very varied kind of skin diseases which affect the skin physiology, including sunburn, which is a kind of first degree it is a sort of first degree burn injury, which mainly affects your epidermis. Then we have acne. So if we talk about skin diseases, it's not just a kind of disease, but I would say it also contributes towards the psychological and the societal effects which are associated with the skin diseases, especially in case of acne, dermatitis, as well as psoriasis. So acne, again, which is caused because of increased secretion of fatty acids as well as formation of comedones within the layers. Then we have some kind of viral infections like herpes simplex, herpes virus, burn wounds, as well as associated infections where certain burn injury could be inflicted with a bacterium, which would further give rise to this kind of infections. Then we have eczema and dermatitis. So dermatitis is a kind of skin condition which is a combination of the environmental factors also, as well as certain kind of immunological changes also which take place within the individual. So basically it is a chronic skin inflammatory condition which occurs because of the changes in filaggrin protein which is present in epidermis. Then we have certain like minor kind of skin diseases, I would say cracked heels, blisters, rash, etc. And again, warts, which is a kind of fungal infection, along with dermatophytosis as well as candidiasis. Then again, some burn injuries, as well as various other kind of infections as well as diseases which may affect the skin. So we can really understand the kind of burden of these kind of skin diseases which we have. Also, because these skin diseases have got a quite different kind of pathophysiologies which are involved. So that is why the kind of formulation approaches which are involved for the treatment are also different. Now, coming over to the nanocarrier approaches which are used for the topical delivery. So in recent times, there has been a rampant use of nanotechnology employing various kind of carrier-based strategies in order to improve the permeation of the drug molecules across the skin barrier. So I would say though nanotechnology has been quite a hype in the recent years, it has been used in varied areas of drug delivery, but definitely topical delivery is one area in which this nanotechnological approach has really made its impact. We can see a number of products which are available in the market, including some vitamin C formulations as well as antioxidant formulations which are based upon these liposome or other vesicular strategies. So this nanocarrier approach has been able to make an impact upon the topical delivery, and also it has been able to overcome the real issues as well as the real challenges which are associated, including poor permeation as well as retention within this skin layers. So if we talk about liposomes, ethosomes, transferosomes, basically these are the phospholipid vesicles and composed of phosphatidylcholine, cholesterol, and an aqueous compartment. And they can be used for the incorporation of both hydrophilic as well as the lipophilic molecules, including having the properties of biodegradability and biocompatibility. So the first liposomal antifungal compound was approved almost about 10 years back. And since then, there has been a lot of research which is going on in this area, especially for the delivery of antifungal as well as antiviral molecules. Then coming over to the lipid polymer hybrid nanoparticles. So this kind of strategy, like a kind of complementary characteristics, combining the complementary characteristics of the lipid as well as the polymeric nanoparticles has also been there. And again, the main benefits which can be reaped off include biodegradability as well as biocompatibility. Then we have the nanoparticulate carriers, specifically including the solid lipid as well as the nanostructured lipid carriers, which not only help in protecting the drug from degradation, but also on the other hand, they provide a kind of occlusive effect, which helps in providing hydration to the skin surface. And this hydration ultimately leads to the permeation of helps to permeate the drug molecule across the skin barriers. Then we have the organogel, microemulsion, nanoemulsion, and similarly a number of other formulations which can be used to deliver these active molecules across the skin layers. So this is a kind of basic research hypothesis or a research plan, which I just thought to share with you all. So those of you who have been working in the area of formulation or even in the area of plant-based research, this kind of formulation approaches can be used in an effective manner in order to deliver not only the molecule, but also some kind of, as we see, there are a lot of herbal extracts coming out, which are having good antioxidant properties, wound healing properties, etc. So these kind of formulation approaches can definitely be explored in order to deliver those kind of extracts which are having problems with their formulation in order to deliver these across the skin. So as I said that there has been an increase in the bacterial resistance, there has been problems with the biofilm formation, and also the currently available drugs which are there in the market, they have got a number of the side effects, I would say, like irritation, erythema, as well as they have got a delayed healing potential. Also, the question arises that why would somebody like to invest in nanocarrier approaches when you have the conventional approaches like creams and ointments available? So the answer is that all these creams and other formulations which are there, they are basically have been prepared with a lot of composition of the traditional excipients. And if you have a look closely, the formulation as well as the penetration characteristics of these formulations are very less. They just form a kind of layer on the skin with a very reduced permeation of the molecule across the skin barrier. So these nanocarrier-based approaches can be used not only to present the drug in a new way, but also a kind of new approach by using the new excipients which are available, including the lipid-based excipients as well as emollients, which help to moisturize the skin as well as provide improved hydration to the skin. So this approach has been used as a part of my examples, which I would be showing in further slides, and that basically involves some kind of factor screening approaches as well as Qbd approaches. Finally, the characterization as well as the evaluation of these drug delivery systems, which can be done by the basic techniques like micromeritics, morphology, drug release, and also some kind of new approaches like cell culture as well as ex vivo studies can be explored in order to really I would say, harness the potential of these nanocarrier-based approaches for enhanced safety as well as efficacy and patient compliance. So since I come from a lab where we did a lot of research on topical drug delivery, and I can really attest with all the kind of formulations which we developed and the work which has been going on in our lab, definitely the nanocarrier based approaches, they provide a great edge to to improving these challenges for topical drug delivery, as well as not only delivering the molecule at the right site, but also with improved dermatokinetic behavior. So I just thought to take this opportunity to like discuss two case studies which I did as a part of my research work. And basically both of these case studies are there in order for the treatment of burn wounds and associated infections. And in this case, two different molecules have been explored with two different kind of formulations. So the first formulation which I would be discussing is the lipid polymer hybrid nanoparticles. And as I discussed, this is a kind of formulation which like combines the complementary characteristics of the solid lipid particles also, as well as having a polymer component added to it. So this was mainly there in order to address the issues of poor permeability, which is associated with fusidic acid. Fusidic acid is a steroidal antibiotic, which is used for the treatment of primary as well as secondary infections. There is a currently available product which is there in the market, but a number of Cochrane reviews as well as systematic investigations which I went through, they definitely indicate that there is a great problem with the permeation of this molecule, and also this molecule is susceptible to degradation by oxidation as well as hydrolysis. So the two major factors to be looked here are, number one, definitely we wanted to improve the permeation as well as the retention capacity of this molecule. And secondly, it was also there in our mind to provide a kind of protection to this molecule so that it is not subjected to atmospheric degradation and hydrolysis. So it was important to reap the benefits of LPHNS, which basically include improved structural integrity as well as bioavailability, along with the polymeric component which was used in this formulation was chitosan, and chitosan has got a great wound healing potential. So it was an aim to also explore this wound healing properties of chitosan and how chitosan can be compared with the lipid nanoparticles for the treatment of burn wounds and as well as associated infections. So basically, these formulations were prepared by hot microemulsion method, and initially solubility studies were conducted in different lipids as well as surfactants, co-surfactants, to find out the correct, I would say the correct ratio, as well as what kind of lipid as well as surfactant excipients can be employed. And basically the primary as well as secondary emulsion method was used in order for the formation of these FALPHNs. So this, the figure on the right hand side, it basically shows the optimization of formulation variables. And this is a kind of, changing one variable at a time strategy. Although quality by design and formulation by design, we have it, and I can definitely say that it has got a lot of real potential advantages over such a kind of approach. However, what I witnessed was sometimes you don't have that much resources, and sometimes you don't have that much time in order to walk around those 15, 16 formulations which are there with the help of this formulation by design as well as quality by design software. So what I did was, in one of the study, I just used this one variable at a time approach, and in another study, I used this formulation by design approach. So sometimes that comes from my experience that this kind of approach could also be used specifically in those cases when you don't have that much amount of time as well as resources in order to prepare those many formulations. And definitely with my results, I was really satisfied, and I was able to optimize this formulation for 5-6 variables which I was having, because there was lipid, there was surfactant, then we had chitosan concentration, and then I needed to optimize the stirring speed. So because of so many variables which were present in the formulation, it was necessarily important to have such kind of optimization. And I prepared like a prototype of 7 to 8 formulations, and then I just had a kind of characterization for these formulations in terms of particle size, entrapment efficiency, as well as polydispersity index, in order to see that which lipid concentration, surfactant, as well as chitosan concentration can definitely give me the formulation which I'm expecting with the desired particle size as well as a kind of high entrapment efficiency. So these were some of the formulations which were developed, and they were characterized with this different techniques, including the Malvern particle size analyzer, which was used, and entrapment efficiency was determined by the centrifugation method. Then coming on after the optimized formulation was further characterized for the surface morphology as well as the kind of appearance. So basically almost three techniques which are commonly used, that is transmission electron microscopy, field emission, as well as scanning electron microscopy. So I did both TEM as well as FECM images of these nanoparticles in order to see whether there was any kind of aggregation or I had uniformly shaped particles or not. Another characterization technique which can be used in such cases includes the FTIR as well as XRD and DSC studies. So I would say they don't provide so much information about the formulation as such. However, they can be a good indicating point in order to see what kind of interactions are going on within the formulation, and also a kind of characterization where you can ascertain whether the drug has been incorporated within the particles or not. So as we see, the in this case, again, I performed the FTIR studies for the pure drug molecule as well as the blank formulation which was there and the drug loaded formulation. So one thing which I came across was sometimes the drug loaded formulation, as in my case, the peak of my drug molecule was missing in the drug loaded formulation, and this could be attributed to the high temperature which was involved in the preparation process, and that could definitely lead to a kind of molecular dispersion of these drug molecule within the formulation. And that could be one of the reasons which, as I went through the literature, I saw that many of the people have reported this kind of behavior, although we might expect that the drug of the molecule would be as such in the final formulation. However, sometimes it disappears too. Then we had the XRD as well as the DSC studies in order to see the kind of again, the incorporation of the drug within the formulation. So these are some of the examples of characterization studies which can be easily done for such kind of nanocarrier-based approaches, in which if you are planning to prepare some liposomal or some solid lipid nanoparticles, these studies can be used as a prototype in order to characterize that particular formulation. And then in vitro release studies were performed by the dynamic dialysis method. And basically, in case of when we are talking about some kind of treatment of burn wound as well as associated infections, it is usually expected that initially there should be some kind of burst release of the drug molecule, so that which can provide some initial drug concentrations at the target site, followed by a sustained drug delivery which can continue for a period of 1 or 2 days or as many days. So in this case, we had a kind of burst release phenomena as well as a kind of the sustained release phenomena, which was definitely good for such kind of treatment of such kind of diseases as well as infections, in which the infection definitely prolongs for a very long period. For example, in case of burn wounds and associated infections, it usually takes a period of about like 13 or 14 days, in which you have to repetitively apply the formulation before a kind of complete remission of that disease can be produced on. Then another thing is in vitro cytotoxicity studies, which I did on human keratinocyte cell lines. So because these formulations as well, especially this solid lipid ones, they involve a number of such surfactant and lipid combinations. So it is very important to understand whether this kind of combination has any cytotoxic effect on the skin cells or not. And for this case, I used the human keratinocyte cell lines, and basically the different drug concentrations as well as the drug concentration which was present in the nanoparticles was tested against these cells. And it was found that, yes, within those concentrations, the drug was not cytotoxic for a period of like 72 hours for which this study was conducted. Then another important thing for topical drug delivery, which has to be considered, is the kind of appearance of that formulation. So although physicochemical characterization of the carrier is important, but it is also important that how your formulation looks and what are the kind of rheological as well as the textural characteristics, which ultimately define the ease of application of that particular formulation on the skin surface. So in this case, the nanoparticles which were formed, they were gelled into carbapole 940, and finally the rheological studies were done using the viscometer, as well as the texture analysis was done in order to define the parameters like firmness, index of viscosity, cohesiveness, as well as consistency. Again, permeation as well as retention studies coming on to the ex vivo part. So this was the major crux where we really wanted to find out if there was any kind of difference which we can see as compared to that of the marketed formulation. And this kind of study was performed in the France diffusion cell using the male vista rat skin. And so you have got a donor compartment and a receptor compartment, and you apply the formulation on the donor compartment, taking out samples from the receptor compartment at different time intervals. And the lab developed formulation performed better as compared to that of the marketed one. And basically, this could be contributed to or the hypothesis could be the fluidization of the lipid bilayer because of the combination of surfactants and lipids which was present in the formulation, and ultimately it helped to move the active drug molecule, that is fusidic acid, across the skin layers. Then after the skin permeation, it was important to identify if certain amount of the drug is retained in the skin also, because the retention is also another important property which is required so that the drug is released from the formulation for a longer interval. And in this case also, the same skin tissue which was obtained from the France diffusion cell was used, and it was found that yes, the amount of the drug which was retained in the skin was quite higher as compared to that of the marketed formulation. Then coming on to the CLSM study, so basically this is a kind of confocal laser scanning microscopy approach where the major thing is just to see is whether these nanocarriers can reach the kind of layer or your target site. It could be epidermis, dermis, or just the stratum corneum. So in this case, the formulation is prepared. So instead of the active drug molecule, the formulation is prepared just with a fluorescent dye, and you perform the normal ex vivo permeation studies, harvest the skin, and then perform the confocal microscopy in order to see the extent of passage of the nanocarrier across the skin. And basically it is a kind of qualitative study which can be performed in order to add up to your quantitative data which you got in the skin permeation profiles. Then the last part of it, coming over to the what kind of models which can be used for the evaluation of this kind of uh, formulation approach. So it is important because there are kind of different models which are available for topical diseases, including like for the psoriasis, we have different models. For acne, we have testosterone induced acne model. And here we have got murine burn wound model, which basically involves investigation of not only the appearance of the skin as well as the histopathological investigation in order to see what are the changes which are occurring at the base level of the skin. So before murine burn wound model, I would also like to put some light on dermatokinetic studies. So basically, this is a kind of prototype which was developed in our lab. And in this, we just separate the epidermis as well as the dermis after the ex vivo permeation studies in order to quantify how much amount of the drug is present in each layer. And it's a kind of approach which is used to further determine the AUC as well as the C skin max as well as the T skin max to find out that whether there are any changes which are occurring in the sort of bioavailability as compared to that of the marketed formulation. So that's another strategy. So usually tape stripping technique is used, which you can mostly find in the literature, but uh, this technique has been kind of tested and proven in our lab since like 5-6 years, and it really produces reliable results. So we have been doing it for a number of studies. And then coming over to the pharmacodynamic model, which was performed. So this is a kind of murine burn wound infection model in which you have mice and you inflict an injury upon the mice. There are a number of different protocols by which you can do this. It can be done with hot water or with a hot rod. And then the second most important aspect in this case was not only the injury, but also the associated infection which occurs after the injury. So in this case, after the mice were inflicted with the burn injury, methicillin resistant Staphylococcus aureus was used as a kind of bacterial infection, and that was placed upon the burn injuries in order to see what kind of effect the formulation would have, not only on the burn injury itself, but also on the level of these bacterial burden. So it was important to not only heal the wound, but also to reduce this the bacterial numbers which were there, which would ultimately lead to the final healing. So in this case, first of all, we performed the bacterial burden studies in which the amount of the bacteria which was present at different samples for different number of days were tested. And then we had some wound contraction studies. So in case of wound contraction studies, you just check out how much amount of the wound has reduced over a period of days. So this, though this kind of healing is a natural process within the body itself, however, the composition of the formulation, including the lipids as well as the surfactants, definitely contributes towards enhancement or as well as the improvement of that particular contraction. So these are some of the images as well as the histopathological examinations which were done using hematoxylin and eosin. And then hydroxyproline content, because hydroxyproline is an indicator of collagen formation. So more the hydroxyproline, more the collagen, and better the healing of the wound. Along with that, antioxidant estimation as well as pro-inflammatory cytokines, because pro-inflammatory cytokines usually during the time when there is an initial injury, there is a rush of these inflammatory cytokines, including the interleukins as well as TNF alpha. So it was important to determine whether at the end of the protocol, so like almost after 10 days, the lab developed formulation could completely reduce that bacterial burden, and there was complete healing of the wounds as compared to that of the marketed formulation, which could do that in like a period of 16 or 17 days. So there was considerable reduction in the time of therapy, as well as we had like just once a day application of this particular regimen as compared to that of the marketed one, which states that it should be applied 2 to 3 days. And moreover, the other thing which we were looking forward was the improvement in the stability. So the stability data also indicate that there was the kind of the loss which was there after a period of 3 as well as 6 months was well within the limits, and definitely the formulation could protect the drug molecule from any kind of oxidation as well as hydrolysis. Along with that, a physical examination of the formulations were also performed. So that was one of the examples, just as a prototype or a case study, which can help you to plan your experiments as well as design your studies, depending upon the kind of equipments and infrastructure which is available at your place. So that is just one example. And coming on to the second example, this is also a kind of nanocarrier-based approach only, but it was used with a different molecule, that is silver sulfadiazine. So as most of you might know that silver sulfadiazine is a kind of gold standard which is used for the treatment of burn wound injuries. There hasn't been any molecule which has been so effective in this the treatment of the burn injuries like silver sulfadiazine, because with antibiotics we have the problem of the antimicrobial resistance, but with this kind of molecule, it usually combines the oligodynamic actions of silver as well as sulfadiazine. However, the problem was, again, as I went through a number of Cochrane studies as well as systematic reviews, it might appear that, okay, there are so many, silver sulfadiazine formulations available in the market, then what's the point to work upon this molecule for topical drug delivery? However, I was also quite surprised to see that the permeation of this molecule is very low, and moreover, this kind of formulation, because cream silver sulfadiazine is usually available in cream formulations, So this cream, whenever it is applied on the top of the surface of the skin, it usually leads to the formation of a kind of eschar, I would say, just as a kind of layer, which further inhibits the permeation of this molecule across the skin. And moreover, in some of the cases, this kind of film which forms on the skin surface, it has even given rise to some kind of bacteria, as well as reduced healing of the wounds. So the aim was not only to have successful entry of the molecule through the skin, but also to provide such a kind of environment to the physiology of the skin, which would contribute towards its hydration as well as natural healing. So in this case, I have employed an excipient that's egg oil, and it has been basically a kind of oil which is composed of omega-3 as well as omega-6 polyunsaturated fatty acids, and it has got great number of the wound healing properties. But mostly it is available as such for the wound injuries. So the plan was to incorporate this particular excipient also as a part of the organogel formulation to bring about that change in the not only in the dermatokinetics but also in the kind of the pathophysiology of the skin. So in this case, as I discussed before, I have used the formulation by design approach. So formulation or quality by design approach. So in this case, first of all, the risk assessment studies were performed. And basically, this is a kind of study which is there in order to identify what kind of risks which might come into the formulation, as well as the kind of the response variables which we have. So for this, the Ishikawa fishbone diagram was constructed. And as you can see, there could be a number of factors, right, ranging from materials to the kind of method which has been employed, to the kind of measurements which have been there, as well as the Poisson errors which could be there, like sampling error, weighing error, as well as mixing errors. So after that, screening design, that is Taguchi design, was there in order to build up this risk estimation matrix and in order to identify that what kind of the critical quality attributes have a high risk factor, which ones have a medium, and which one have a low. So these were the critical quality attributes which were there, and then the response variables were there, including viscosity as well as physical stability. So initially the formulations were tested just for these two response variables, and with that we had a kind of 7 type of prototype formulations which were prepared and then which were further analyzed by this Taguchi design. So, and this formulation involving phospholipid 90G as well as some surfactants was basically prepared by a simple mixing method, which ultimately resulted into the formation of a 3-dimensional network of entangled micelles. Then the factor screening studies were there by viscosity as well as physical stability, and then all those factors which had the values above the Bonferroni limit. So they were chosen for the final systematic FPD optimization as per the optimal mixture design. So in this case, for the optimal mixture design, PL surfactant and the ratio of the concentration of the surfactant as well as the concentration of ethanol and other components were optimized in order to have their effect on drug release, particle size, viscosity, and phase transition temperature. And then in this case, optimal mixture design was used. And finally, using response surface methodology, we could we could land up to a kind of optimized formulation, which had the maximum drug release as well as the desirable viscosity as well as PTT, the phase transition temperature. Now, again, these formulations were characterized by microscopy, in this case, optical microscopy, as well as the TEM image method was used in order to see the kind of the micelles as well as the kind of uniformity which they are having. Again, as I discussed previously, Clsm studies were there. So this can be a kind of good study which can be used just for the qualitative assessment that whether the formulation has got a penetration capacity or not, in addition to the skin penetration studies which can be performed with a France diffusion cell. Then again the FTIR studies, again this could be performed with all of the excipients or with just the blank formulation, the drug loaded formulation, and the drug in itself. Then ex vivo permeation and again skin safety studies. So in this skin safety studies, basically the idea is to have an initial application of the developed formulation on the skin for a period of like 6 or 7 days, and to have a kind of physical assessment by visual inspection to see what kind of changes occur. Is there any kind of inflammation, erythema, redness, etc.? And secondly, these skin samples can be easily harvested, and then the microscopic images could be obtained in order to see what kind of changes are taking place in the formulation treated skin as well as the comparator which we had. So it was also in this case we had a marketed formulation in order to compare. Then again, dermatokinetics, which again provide a useful information about how much of the drug is available in epidermis as well as dermis, because sometimes for certain molecules as well as certain skin conditions, the desired target site is epidermis, and on the other hand, it is dermis. Then again, pharmacodynamic studies involving determination of bacterial load, wound contraction, hydroxyproline, as well as the histopathological examination. So these were two examples from the kind of studies which I did. However, there are different number of formulations which could be prepared depending upon the physicochemical properties of your molecule, including liposomes as well as ethosomes, transferosomes. They have been used widely for the delivery of such kind of active molecules. And also there could be different pharmacodynamic models which can be used in order to see the therapeutic efficacy of these formulations. So those were the two case studies as well as examples which I wanted to discuss. Now this one is so this work was done at University of Nottingham, and basically it involved preparation of some kind of biomimetic scaffolds for the wound healing applications. And here the idea was to sustain the delivery of the drug for a very longer period of time, maybe like about 30 days or 40 days, depending upon the kind of the polymer which was used as well as the kind of its release properties. So basically in this case, electrospinning technique was used, and electrospinning technique mainly involves usage of the electrostatic properties of the polymer as well as the solvent which is used. So you've got a polymeric solution within the syringe, within this equipment, and then at an specified electric voltage as well as flow rate and pump motion, you have these fibers collected over here because of the electrostatic interactions. And this is the kind of the scaffold which you have collected on the aluminium foil. Further, the scaffold can be easily removed from this aluminium foil, and it can be further characterized for different kind of studies. So scaffolds is another area which has been developing very quickly. However, there are certain, I would say, restrictions or inhibitions which are associated with the use of the scaffolds, because a number of studies have also stated that scaffolds not only deliver the drug molecule at the target site for a sustained period, but in certain cases, these scaffolds have also been known to cause some kind of biofilm formation or some kind of emergence of the bacteria within the space which is present between the scaffold as well as the wound. So still this work which I did was just for a short period of time. I didn't had so much time to explore it. However, I could really have a gist of how these scaffolds could be prepared by different kind of methods, and what could be the factors which are important for the optimization of these scaffolds, including concentration, conductivity, viscosity of the polymeric solution, as well as a number of the other process parameters. So not only polycaprolactone, but a number of natural as well as synthetic polymers like chitosan as well as the PEO have been used in order to design such kind of scaffolds, and they have been used for the delivery of antibiotics as well as a number of other anti-infective molecules. So this is a slide stating about the difference in the blend as well as the coaxial scaffolds. So in case of the blend scaffolds, the strategy could be to incorporate just one kind of molecule within the scaffold. However, in coaxial scaffold, we can always have a kind of balance between the hydrophilicity as well as the lipophilicity. So you can incorporate a kind of synthetic polymer which degrades at a very slower rate. On the other hand, there could be something like polyethylene oxide or any other hydrophilic polymer which can further fasten this degradation rate. And then the scanning electron microscopy as well as transmission electron microscopy, these are some of the other techniques which are used in the initial characterization of these kind of scaffolds. And further, I haven't shown up the slides here. However, the major work in such kind of characterization of scaffolds basically involves the cell morphology studies which are carried out on the scaffolds. So scaffolds, they are incubated with cells to see that what kind of contribution they have towards the cell culture experiments. So that could be another set of experiments which are performed for these blend and coaxial scaffolds. However, these kind of wound dressings, I would say, because of the restrictions which are associated with such kind of drug delivery system, they haven't been although they have been commercialized to a great extent, but still there are certain problems and there are certain discussions which are going on about this kind of drug delivery platform. Now, coming over to the formulating for efficacy software. So this is one of the software which I worked on in recent times, and I thought it would be a good point to discuss and to share this thing with you all. So what we see is that when we prepare some kind of topical formulations or for the formulations which are meant to be delivered across the skin, obviously there are a kind of number of penetration enhancers, there are a number of emollients which are available. So solubility studies can definitely be used in order to determine what kind of penetration enhancers and what kind of surfactants and emollients would be useful. However, sometimes it is not feasible to conduct, solubility studies at such a great level in order to determine what kind of, proper matchup of those surfactants and emollients would be suitable for your kind of formulation. So this is a software which helps you to determine not only the concentration as well as the type of the emollients which can be used to deliver your or to dissolve your active ingredient, but it also helps to take things into account from the physiology of the skin. That is, what kind of emollients would be best suited for the permeation of these drug molecules across the skin. So basically, it's a computational kind of software in which they have got a database of about like 100 to 100 emollients, and if the desired emollient which you want, it's not there, you can enter it. You can enter the active ingredient, and then you need to have information about the oil water partition coefficient as well as the concentration of the active ingredient which you are looking at. So, and then further thing comes up to the optimization, that is what kind of strategy are you looking at. Either you want to increase the solubility of your active ingredient, which is very much poor, or you want to improve the penetration of that particular active ingredient against the stratum corneum, and thirdly, the concentration of these emollients. So this kind of software, it can really help to answer many of the questions which we usually have when designing such kind of topical formulations. And obviously you need certain kind of experiments in order to calibrate the kind of data which is obtained. And also it can also not only be helpful in just the designing stages, but also in case when you have a kind of formula and you really don't want to add any new ingredient to your formulation. So in that case, the software can be used to optimize the ratios of existing ingredients depending upon, again, what kind of objective you have, whether you want enhanced permeation or whether you want enhanced solubility of the drug molecule. Then If you need to add one ingredient, or what kind of ingredient you have been looking at, whether it is a kind of penetration enhancer or you want a combination of some penetration enhancers as well as emollients. So in that case also, it helps to identify the best new emollient as well as its required concentration. And then again, if you want to add some like 2 or 3, then again, it could be really helpful. So although it really doesn't solve all of your problem, but I would say it's like, reaching at least at the base of the Himalayas, you will have a definite idea about what's going on and what kind of combination of the emollients can definitely solve your problems, because there are like so many types of emollients available, penetration enhancers available. It's not always easy, to conduct solubility studies in like 40s or 50s of these before carrying out an experiment. So definitely this could aid into that, and this could help into that, and this is available on the jwsolutionsoftwares.com. So if anybody wants to have a look there, you can have a detailed look at that, this site. And also there's a kind of demo which is available, so it might be useful to many people. Then the last slide. So that's my current area of work, which I have been working. Earlier, I was working in the formulation scene of things, but now I have moved to the PBPK modeling and simulation and drug development. So it's not a new concept, I would say. It was started in the year 1937, but it has now started getting approval from federal agencies just in the last like 5 to 10 years. The number of approvals which have been for the generic drugs on the basis of PBPK modeling and simulation has been increasing at a tremendous rate. So basically, if we talk about classical pharmacokinetics, in that case, we assume that the drug is uniformly distributed throughout the body, and the elimination of the drug could be defined by a rate constant, which would stay same. However, that is not the case. Most of the times, the physiology of the body, including the blood flow, the tissue composition of the organs, as well as the abundance of the enzymes and the transporters which are available, it really has an effect to define the pharmacokinetics of the drugs. So it is a kind of computational modeling as well as, I would say, mathematical approach in order to take into account all these factors. So you can have an account of the physiology of the body, as I said, including the enzyme as well as the transporter abundance. Then you can have an account of the physicochemical properties of the drug molecule, like log P, partition coefficient, solubility, etc., pK, etc. And then the other thing is you can have an account of the demographics of the population. So the age of the population, the weight of the population, as well as if you are looking at pregnant population, if you're looking at children or the elder people. So in recent times, this kind of approach has been saving lots of money for the pharma companies, because sometimes it is not easy to conduct kind of clinical trials in sensitive population, including pediatrics, pregnant population, etc. So this kind of approach can definitely, help to wave off many of the clinical trials, and it has already helped. I have been working currently in the area of oral PBPK modeling as well as the dermal approaches which are used in drug development, where we take into account all these kind of physiological as well as drug related and trial design related aspects in order to make some predictions which can definitely help to wave off the clinical trials, and thereby it can contribute towards the drug development as well as an approval process at a very fast rate. And specifically, I would like to mention for topical drug delivery, this has been a kind of it has got a kind of recent very much attention has been there by the FDA for PBPK modeling and simulation approaches in order to fasten up the approval process for the generic drugs, because a number of topical products which are available in the US market, so they have got generics for just 2 or 3, which ultimately adds to the cost of these products. So these kind of approaches have been used along with a kind of pharmacokinetic studies, but with a less number of patients and less number of clinical trials in order to contribute to this fast approval process as well as the development process. So that's one of the emerging areas. And if anybody wants to have a look, there are like number of literature articles and papers and chapters which are available, and also there are a few softwares also which are available, and you can have a look at that. And yeah, so with this I end my presentation, and I would extend my warm gratitude to everybody for a patient hearing, and I would welcome if you have any kind of questions, comments, suggestions, or any thoughts from your side.
Thank you so much, ma'am. A great session, I must say. Dr. Anoop has a query which he has already posted in the chat box, and I would request if he could ask it directly to ma'am. Dr. Anoop?
Hi,
Yeah.
yes, ma'am. Hi, ma'am. Very good afternoon, and very nice work you have done and doing. I have one query related to toxicity studies of topical formulations. If suppose any topical formulation is able to cross the skin barrier and reach into the systemic circulations, then in that case, we have to do systemic toxicity studies or not.
Okay, so first of all, thanks a lot, Dr. Anoop, for your question. So yes, there are certain kind of drugs which can cross the skin barrier and they can enter into the systemic circulation. And I have dealt with one of such molecules. And in that case, I would say, yes, it is important to have an initial blood level studies in order to see what kind of, concentrations are there in the blood for that particular molecule. And whether they are in the permissible limits or there are like some possibilities that it can contribute towards toxicity. So in that cases, definitely I would say that conducting systemic studies, at least an initial blood level PK study, would help in having an initial idea about that.
Okay, ma'am. Thank you, ma'am. Ma'am, another query is related to if we are doing a toxicity studies. So means, is there any standard how much surface area we have to be cover for the topical applications? And
Okay.
how to calculate means how to decide the dose in that particular area?
So I would say that in that case, most of the times it's usually almost about 5 milligram per centimeter square or 10 milligram per centimeter square of the area is generally used for such kind of toxicity studies when you're doing it on the surface of like skin. But on the other hand, you can also have a kind of calculation on the basis of amount of the formulation which is recommended. So if you have an idea about how much of the dosage that is required to be applied, it is definitely, it would be good to have this 10 milligram per centimeter square within that range. So you have to calculate and you have to approximate the amount you want to apply accordingly with the kind of dosage which is prescribed.
Okay, ma'am. Thank you very much, ma'am.
Thank you. Ma'am, Vandana Patravale ma'am has specially mentioned, if you can see in the chat box, excellent presentation and work, Kanika.
Oh, thanks a lot, ma'am. It's really a great pleasure, I would say, to have these comments from your side, because I mean, I would express this with all that I have been, during my PhD time, I have just gone through so many papers of ma'am, and I'm really in great admiration and respect for her. So, it's a kind of blessing that we have such kind of people in the pharmacy profession. Thanks a lot,
Absolutely.
ma'am.
Absolutely, ma'am. Dr. Sibi would like to ask a query. Dr. Sibi?
Good
Sir,
evening.
yes.
Good evening, madam. I'm having some doubt in that first model, pharmacodynamic model, which you have mentioned, murine burn wound model. Normally, this is a cruelty to the animals, and the institutional animal ethical committee, normally they are objecting this type of cruelty to the animal models. So how we have overcome that objection? Another query is related to that infection, injury and infection after the burn with the hot water. You told that there are some chances of getting infection. So especially the methicillin resistant bacteria and all. So whether we are along with your product, whether any other antibiotics or any antibacterial compounds were administered or not. If it is given, then naturally the results will be, these adding agents also will have an impact upon your product also.
Okay, so thanks a lot, Dr. Sibi, for your question. Coming on to your first question, yes, I would agree to that to some extent that yes, I wouldn't say no, it is a kind of cruelty, but that is something which is, we don't have any answers in our profession. Definitely, if we want to have, the extrapolation of this lab work into clinical studies, we really need to get our hand, at least initially, on this animal models and pharmacodynamic models. So for that, I got the ethical approval from my university, and we have a kind of, definite meetings in which you have to propose the kind of plan and the kind of animal model which you want to do. But the thing that was taken care of by me was to have least number of animals which I can have in a particular group. And yes, I would say that it is somehow difficult in order to carry out these experiments also, and considering on the basis of, as you said, cruelty. But then I would say the entire pharmacology division would be just gone off from the scene in case, if we think about this, if we really want to have good therapies for, at least for the human species, we need to do this. So that is my take on that. I mean, yeah, everybody has a different opinion, and I would respect that too. But that is my say, which I would say that it is, indispensable to remove these kind of experiments from the preclinical stages, which are done if we want to have an idea about how the formulation and how the molecule is working. And coming on to your second question, that is, yes. So initially, the burn wound injury was created by a kind of a hot iron rod. So in this case, hot iron rod was used, although hot boiling water can also be used. And after that, a kind of inoculum of MRSA bacterium was added into the burn injury. So yes, if we have additional antibiotic formulations, that effect would contribute more. But in this case, the molecule which I was using, fusidic acid, is an antibiotic in itself. So it was obviously the property of the molecule also to inhibit the bacteria. But secondly, the kind of delivery environment which the molecule got also added towards the enhanced as well as faster reduction in bacterial burden as well as wound contraction.
Okay. Okay, madam. Thank you. Thank you very much.
Thank you.
Anyone else who wants to ask, ma'am? Anybody? Well, I guess no one else. Ma'am, I would like to share my screen. If you could kindly stop the sharing.
Yeah, sure.
Thank you, ma'am. So, ma'am, thank you so much once again from Lloyd Institute, and kindly accept this e-certificate from our side. Thank you so much.
Thanks a lot, Dr. Shilpa. Yeah, it was a pleasure to be with you all. Thank you.
Same here, ma'am. Thank you so much. So we'll meet again tomorrow at the same time, 1 p.m. Thank you and bye-bye.
Thank you.